severity, it was observed that Vanguaria rotundata was actually, the incidence and severity was high in Vanguaria as compared to Bachamia discolor in all the sites, but the infections were actually more severe in Tiva as compared to other sites like Ikanga and Mkange. Then morphological characterization, at least 7 fungal families were identified morphologically, with the most dominant being Protosphaeriaceae, followed by Dectriaceae and Sporocardaceae, Pleosporaceae, Glomerulaceae, Trichosphaeriaceae, and Diapodaceae. These are some of the images of fungal identification morphologically. And these were the most dominant fungi that were isolated. There was Protosphaeriaceae, and you can see the color of the basilia, and also how the spores are ellipsoid, a bit opaque, and with one septa. There is that of the Sporocardaceae, where you can see the white mycelia with a bit of pycnidia, black pycnidia, and the spores were hyaline with at least 2 to 3 appendages and 1 to 2 septas. And the characteristic for the Nectraceae was that the mycelia was white, and the spores were a bit curved with 2 to 3 septas. DNA analysis. This is where I actually did the editing of the sequences, where I had to truncate the sequences where the noises were a bit too much, and also correcting some of the miscalled nucleotides. And then I was also able to compare the sequences from my fungal isolates with those from the NCBI database, and they were assigned names according if the similarities in the nucleotides was between 99 to 100%. And then the sequences from the isolated fungi and those from the NCBI database were aligned. I did a multiple sequence alignment, and from that I was able to generate a phylogenetic tree, in which my isolated fungal DNA sequences were able to cluster with those of the and phylogenetic tree, and you can see that the fungal isolates grouped into the 7 fungal families that I talked about: the Nectriaceae, Glomerulaceae, Diapodaceae, and Trichosphaeriaceae, and the fungi with that clustered with the sequences from the database with 100% bootstrap support, I was able to actually conclusively identify them. However, there is a family that I was not able to identify, like any fungi to the species level. And that is why it is encouraged to use multiple gene regions for identification. Next slide, please. The pathogenicity test was carried out using the average number of days that it took to show the disease symptoms, as well as the internal lesion length that was the mean internal lesion length. And this showed that Lasiodiplodia lignicola, Anaphilipsia allogenea, and Dodierera were the most virulent on the 3 species, and they were also able to cause similar disease symptoms on the trees that were used for the pathogenicity test. So the conclusion is that Botrytis are opportunistic pathogens, and they cause diseases on a wide range of hosts. And in this case, they were isolated in both from the diseased trees, hence they were considered to be the leading cause of the caca and tobacco disease infections. And through the pathogenicity test, it was clear that indeed that the species from the Botrytis family in the genera Lasiodiplodia and Amphilypsia and Dodierella were actually the potential pathogens that were causing the canker and dieback diseases. This study also emphasizes on the use of molecular methods for the identification, since it is rapid and precise, unlike morphological methods that were laborious and time-consuming. And also emphasizes on the need of accurate and timely and early detection of diseases in not only forestry, but also agriculture and horticulture sectors. So recommendations is that there should be regular monitoring and reporting of diseases for timely management. We embrace the advanced molecular tools for pathogen diagnosis, also including the use of AI, where we have so many diagnostics app where farmers can actually be able to detect and also be able to tell what is actually causing diseases in their farms. This study also encourages genetic diversity and population structure to be undertaken for the 2 tree species. Sequencing of the genomes of the fungal pathogens to identify the gene regions involved in the virulence, and also conduct comparative genomics to find virulent genes that are conserved across the different fungi. So it also encourages the use of the multiple gene phylogeny using either other gene regions such as elongation factor 1, or beta tubulin for identification of some of the species that were not conclusively identified. Thank you.
Thank you, Susan, for your presentation. Is there any question for Susan? No, no question. I have a question, Susan. Oh. Okay. I wanted to ask Susan, the places where you've sampled the 3 species, do you have did you kind of the environmental aspect of the different places? You say you sampled in 3 different locations.
Yes.
Okay.
Yes.
Is there any similarity? What can be the confounding factors that can lead to the presence of these 2 fungi in those different places?
Okay. Thank you very much for the question. The 3 sites were actually in the agroecological 4 and 5, which are characterized by reduced rainfall, high temperatures that actually predispose the 3 species to infection. I said that the Botrytis family normally attack or cause disease on trees that are already stressed. They may exist in the tree as endophytes, but they may take a long time before they cause disease symptoms. But once the tree is stressed, then they are actually they predispose the trees to infection. In the infections, we are also high in TIVA as compared to the other sites, probably because TIVA was actually the plantations and the farms were actually the source of planting materials. So I think the infection may have spread through the distribution of the planting materials from TIVA.
Okay. Thank you very much for your answer. And let's give a last round of applause to Susan, please. Now we move to actual today session because Susan was one of the two remaining that was to present yesterday because of very interesting sessions yesterday. We didn't manage to finish. Okay, there is another question. Yeah, morning.
Yeah, we recognize that. We are fitting you in today's program, but we have people who are online, so we want them to present and then you can present.
Thank you. So we'll start with today's session and we will start with Dr. Andrew, who is a research manager at Center for Ecosystem Restoration in Kenya and specializes in restoration ecology. Andrew has a PhD in botanic, conservation genetics, and at his current position, he focuses on conceptualizing innovative ideas aiming at ecosystem conservation. He also works with international partners to develop and adapt global biodiversity standards. And he's also published, he has a very long list of publications from various areas, ecology, botanic, molecular biology. Well, if you want to know more, you can visit his LinkedIn. He has quite a long. Andrew, you have the floor, please.
Yeah, thank you so much, Dr. Mendez. Yeah, good morning, everyone. Yeah, and those who are online, I'm sure we are in different time zones. So hello, wherever you are. So I'll be taking you through this talk. It's more of an awareness creation and highlighting some of the challenges that we've been facing as an organization. So the Center for Ecosystem Restoration, its main role is just to start to restore biodiverse ecosystems within Kenya and support similar initiatives elsewhere. So I'll be taking you through enhancing how we've been enhancing biodiversity in restoration projects and how we can adopt global standards to make sure that we're meeting the desired goals. So, nice, sorry. Nice, yeah, I'll start with this slide because we are here now, and I am sure that we've talked about different zones in the past. And Kenya is a very beautiful country, as you can see. And this beauty comes with its biodiversity. We have these different biomes within the country. And there are many, there are more than that, because if you look at this map, we have different vegetation zones classified from K1 to K7, right from the flora of tropical East Africa. And then the different colors on this map indicates different landscapes or ecosystems, running from Afro-alpine zones all the way to deserts and coastal ecosystems. So it's quite biodiverse. And what's happening now is that we are losing this at an alarming rate, because at the moment, 60% of this, the total land area in Kenya is degraded. And actually 27% of the same land size is highly degraded. Okay, now it's clear. And with this land degradation, what's happening, we are not losing only land and habitats. Every moment we lose habitats, what's happening then? We are losing biodiversity, again, which has been quite accelerating. And this is despite the many or the numerous efforts, conservation efforts that are being put in place. And even data is indicating that within this century, we might lose 40% of biodiversity in key taxonomic groups in certain habitat types. And if you ask where this is happening at, or what's the main driver of this biodiversity loss, top on the list is land use or sea use change, followed by unsustainable extraction of resources, pollution, invasive species, and such. And then with this in mind, over the years, there have been a lot of initiatives and conventions, and the most key, I think, and quite relevant to this conversation we're having now is the CBD, which aims to conserve biodiversity at multiple levels, speaking of molecular level, species level, and even up to the upper level of biodiverse ecosystems. Sorry. And with that, with those conventions, there's always these commitments, global commitments, which again, starting from 2008 with REDD+, where forest restoration was adopted, and then in 2011, where we saw Bond Challenge that aimed to restore degraded landscapes with an objective of restoring about 150 million hectares at that time. This has been changed and revised, but that is at the global landscape. And with those major initiatives or global initiatives, we've seen countries committing to do the same, restore and mitigate, stop biodiversity loss within their boundaries. This is the journey that Kenya is aiming to go through, committing to plant about 15 billion trees and restore 10.6 million hectares of degraded landscapes and rangelands by this timeline within the next few years. And these are the targets in the global biodiversity framework. Doing or working towards this journey here, the government aims to meet directly and indirectly nearly all the other targets. But these are the ones that I chose because we are contributing or supporting government directly in meeting them. And particularly target number 4 aims to stop species extinction and conserve biodiversity or genetic diversity within the ecosystems. And within the country, again, globally, looking at the global perspective, we have about 58,000 trees that have been identified. And I focused on tree for this talk because these are the go-to species or taxonomic group when you want to restore ecosystems. In most cases, we'll see whether that is the right decision or not. But then, out of those 58 or nearly 58,000 trees, 84% of those have been assessed. Their conservation status is known and their threat level identified, and 32% out of those have been identified to be threatened globally. And when you come to the country level, we have about 1,118 tree species. These are native to Kenya. And out of those, it has been identified that 44 of those are threatened and 47 are actually endemic to the country. And this is important piece of information because then these are the species when it comes to ecosystem restoration, these are the type of species that are less likely to naturally regenerate in the wild, and they need a lot of support during restoration. And therefore, we'll be looking at these 2 categories in the next few slides. And this is what is happening now. Having identified the challenges, we are losing biodiversity. Land degradation is peaking, is at a faster rate. So what's the solutions? And are those solutions that you're proposing and even implementing and adopting, are they working to reverse this trend? So there are numerous articles answering this question. And in most cases, you'll find that most of them are pointing to the opposite direction. You're not meeting these targets. So I picked only these 2 just to mention that even when we know that trees are the go-to solutions to land degradation or to restoration, they do not always meet the desired objectives. And this is because tree planting is not a very simple solution. And we'll see why. And then there's an article here that assessed what's happening with the commitment that the government have made in Africa. And actually, data collected through this article indicated that about 52% of tree planting is happening in the wrong places in the savanna, where you're supposed to be maintaining this the status quo and supporting the natural balance that is there between grasslands and woody vegetation, but that's not happening. And even worse, most of those 60% of those projects are restoring ecosystems using non-native species. And from the story that Dr. Cecilia was giving us here about biodiversity, maintaining that native ecosystems in their natural status supports even other species in thriving and surviving. And therefore, just to support, is there a way that then we can award and recognize those projects that are doing the right thing, planting trees at the right place, and the right tree, having the right tree, just to make sure that we are supporting the development of these ecosystems. So this consortium of renowned experts in biodiversity, ecosystem restoration, and land management came together and decided to develop this global biodiversity standard where we are assessing ecosystems of restoration projects and rehabilitation projects and promoting or recognizing those that are making positive impact in restoration. So on the right here, you'll see those technical partners that supported this initiative. And on the right here, you'll see the hub model that has been adopted to trial this methodology and make sure that it's pinned in scientific principles and that it can be replicated in different parts of the world, just to make sure that there's that standardization around the world. And yeah, this is the organization that we work with here in Kenya. And the principle behind, or the concept behind the global biodiversity standard is pinned down into these 8 criteria of assessing projects. And these were adopted from another article that was published in 2020. That was seeking to bring about 10 golden rules in reforestation, or looking at different aspects of holistic restoration projects. So when you look at these 8 criteria, I would like our attention to focus on these 2 here, because the criteria number 4, although they are all quite important and you cannot achieve the desired objective without having accomplished the other, criteria number 4 aims to assess projects and to ensure that they are aiming to maximize biodiversity within biodiversity recovery within the restoration sites. And then number 7 here looks at promoting biodiversity and adaptive capacity within those projects. These were now molecular, or that species level of variation comes into play. And it's one of the throughout our assessing, as we were trialing this methodology, this was one of the areas that was quite challenging to assess in the field. And I'll explain why. You cannot assess genetic diversity in the field just by looking at them. So in the last 2 years, we've been assessing different projects within Kenya, about 18 of them in the country and 1 outside of Kenya. And these are spread out across different land use systems, from projects that are going on in protected areas, but they are doing some form of restoration. For example, in forest, where KFS is the forests that are being managed by KFS, and then in agroforestry systems, you mentioned about coffee and monocultures in those areas and whether they are supporting biodiversity or not. And then in restoration areas, like in the project that we run at Bracken and Limuru. So from these projects, then we get different perspectives, and you can understand the challenges that organizations are facing at this level, trying to implement. There's a lot of information out there, but when it comes to the ground to do the actual work of ecosystem restoration, then you meet a lot of challenges. And part of it is because there's no that rapid flow of information. So what we noticed is that those research articles that have been published are quite correct, and the data that we were collecting actually aligned or supported their conclusion that most of the restoration work being done in Kenya is not meeting this global standard. And actually diversity, most of them are using ecosystem native species, and these have a negative impact on local and native species, bringing that out, that competitive aspect. So one of the key attributes that we've been looking at is species composition. And even in projects where you find that there's a lot of species being integrated in the restoration project, you find that there's also, they lack in functional, other aspects of diversity, functional diversities, and even, functional diversity is missing. But just to emphasize the use of native species here and the negative impact that they have on the survival or the competitive trait in the native species. So, and we looked at the challenges. We've been looking at the challenges. Why are these projects not meeting the desired objectives of increasing native diversity in the restoration project? So we assessed about 44 species at the coastal region, and these are tree nurseries. And these are tree nurseries that are linked directly to restoration project. So they're supplying to them with a propagation material. So you realize that out of the 297 species that are being propagated at the coast to support restoration project, almost 75% of those are exotic species, and 69 have been assessed, as listed here, as shown in this graph, and the native species, about 51 are threatened with extinction. So, and then there's another question that came up at this point: why are these nursery attendants, why are the nursery managers not using or not maximizing biodiversity, native biodiversity in the ecosystems? And the question that, the answer that they were giving, most of them giving, that there are these gaps in identifying and knowing even which species are native species, because these are the people who are going out collecting seeds in the wild and also propagating them. So whenever they encounter challenges, even in propagation techniques, they just tend to go to the tree species that are quick to germinate. So we did another survey with restoration practitioners at the coast, and just to identify challenges that they were meeting. Top on the list was lack of skilled labor. Again, we've been talking about capacity building and strengthening the infrastructure in ecosystem restoration. So with this the size of this circle indicates that this was the most limiting factor in the aspect of human resource. Also land and water supply at the coast, and funding environment was also another significant limiting factor. So these were the four at the top of the list, the four main challenges that these organizations were meeting and limiting their capacity to address or to increase biodiversity in restoration project. So, and then we've been mobilizing, having known that, what we've been doing now is conducting training in different aspects, in different parts of the country. This is a project one of the projects that is focusing on doing that, or building capacity in the highlands, or strengthening capacity in the highland zone, K4 zone, vegetation zone. And what we've been doing is supporting them with understanding species mapping, just so they know where, or to come up with seed collection zone maps. And then they'll be in a position where they can be understand how to increase or to protect and even promote genetic diversity, even without that knowledge of having to do molecular sequencing in the lab. And this is mainly based on the data that has been published, again, from articles or from work that has been done in the laboratory. Also understanding population dynamics of species, the minimum number of species that you can, or seeds that you can collect from a single mother plant, or even from a single population, having in the mind the understanding of how to crossing the population biology of this, monitoring phenology in this era of climate change. Most of this understanding when these native species pollinate or produce flowers, and when they produce seeds, because most of them, they have like recurrent seeds, most of the threatened tree species. And therefore understanding when to collect seeds and how to process them promotes the chances of increasing their numbers in the nursery, in the tree nurseries. And out of that, we've realized identifying these threatened tree species within this region, we've been able to develop propagation protocols, even for the most critically endangered species like these Euphorbia cosonoides. And this knowledge is now being transferred to the nursery attendants and all the way to the restoration practitioners on the ground, just to ensure that we are supporting not the recovery of these threatened tree species and not losing this biodiversity. And also in the aspect of seed collection, partnering with international organizations to obtain seed banks, and then conducting this seed collection training and developing seed collection network, community-based seed collection networks from different provenances for each species, has also supported diversifying the number of species or native tree species in the nurseries, and also ensuring that we maintain the genetic diversity of the rare and threatened tree species. And lastly, the takeaway here for us in this room is that there is an urgent, we realize that there's an urgent to address biodiversity loss at all levels, running from understanding the genetics, species, and even that landscape level. And then lastly, and then secondly, there's that importance of adopting global standards in biodiversity conservation and restoration, just to ensure that we are meeting, or we are dealing in an effective manner with three threats of global biodiversity loss, climate change, and livelihood, loss of livelihoods. And lastly, promote participatory and incentive-based conservation to address challenges. This is what has been missing also on the ground, having that incentive-based and providing alternative sources of income or sources of livelihood for the local communities to desensitize bad practices in restoration projects. These are our partners. From a research point of view, we've been working with JQuat in monitoring biodiversity in the different landscapes we're working at, and also formed a consortium between these three international universities. For seed training and collection, we're working with Kefri locally, and most of these projects are funded by international partners. Thank you so much.
Thank you, Dr. Andrew, for your presentation and bringing this kind of discussion in the room. Most of the time, conservation, when the program to put in place restoration, soil restoration happen, most of the time, all factors are not being considered, especially the soil biodiversity or soil ecology. And it is interesting to hear such a presentation. The next presenter will be joining us from online. She's Mrs. Catherine Mungai. Is she connected? Okay. So Catherine Mungai is a senior program officer at IUCN. Okay, I'm pronouncing in French, but in English is IUCN. So I hope that's correct. Where she provides policy in the IUCN in Kenya. Yeah. So she provides policy, advice on aligning climate and biodiversity management with economic development and private sector. So Catherine is the local focal point for BioDev 2030 initiative, which encourage ambitious science-based commitment through voluntary contribution. And she's also an alumni of DAAD, which where she got her master in environmental governance from Freiburg University in Germany. She's passionate about integrating gender and youth into science policy. So Ms. Catherine will present on the role of IUCN in the implementation of global biodiversity framework. So, Ms. Catherine, you have the floor.
Hello, good morning, everyone. Kindly confirm you can hear me.
We can hear you.
Okay, thank you. Thank you very much. Thanks a lot for the great introduction. Yes, my name is Catherine Mungai, and I'm a senior program officer. I focus on climate change and biodiversity engagement at the International Union for Conservation of Nature, and I'm based at the Kenya country office. And having listened to the presentations yesterday and also today morning, I feel a little bit intimidated, but also very pleased because I think my presentation would also be very interesting in terms of providing the science and policy linkages, especially based on all the interesting findings and all the interesting work that is being done in the country and also in the continent. So as we move to the next slide, I will just, my presentation will be based on what we do at IUCN, but also within the framework of the Global Biodiversity Framework. And kindly move to the next. Yes, thank you. So I would like to start by setting the scene. When we talk about the Global Biodiversity Framework, where did this all originate from? And because today we are talking about science and also linking science to policy, I just picked a few facts from the work that has been done by IPBES. And IPBES is the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services. And these are just some few facts to set the scene for my presentation. For example, we see that from the 1900s, the average abundance of native species in major land habitats has decreased by over 20%. And I think this also links to some of the presentations that we've had this morning and also yesterday. We find that over 680 vertebrate species have gone extinct. The IUCN Red List reports over 41,000 animal species are at risk of extinction. We also have direct drivers such as pollution, land use change, exploitation, and climate change, alongside indirect drivers like societal behaviors have led to increased threat of biodiversity. And then progress towards achieving the UN Aichi biodiversity targets in 2011 was limited. And at the same time, we find that the natural ecosystems have diminished by 47%. And this has led to the loss of 120 million hectares of forests. And I think this links to the presentation that we just had from Dr. Andrew in terms of what do we need to restore our forests. So based on those, on that background, countries have come together in 2022, and they've adopted what is now being called the Kunming-Montreal Global Biodiversity Framework. This is at a policy decision that has been made at the global level, but it has relevance to us at the African continent and also at the national level. And we find that the response to the global assessment, this is in response to the global assessment report of biodiversity and ecosystem services that was done by IPBES. And the idea of the Global Biodiversity Framework is to provide biodiversity policy and action, which is guided by 4 goals, which you'll see in the next slide, and also 23 targets, which need to be achieved by 2030. So when we move to the next slide, we can see the 4 goals. The first one is about protecting and restoring biodiversity. And this is the need to ensure the integrity, connectivity, and resilience of all the ecosystems are maintained. And the second one is on ensuring that we prosper with nature. So this is about sustainable use of biodiversity and ensuring that nature's contribution to people and even ecosystems functions and services is well maintained and enhanced. Then, of course, the third one is on benefit sharing, which needs to be done in a fair manner. That's the monetary and non-monetary benefits from the utilization of genetic resources and the digital sequence information on genetic resources, as well as of traditional knowledge associated with genetic resources. And then finally, the goal D is focusing on the need to invest and collaborate. And we'll see in the coming up slides and activities, there's a lot of need to raise finances. But apart from that is the issue of capacity building, which is both technical and scientific cooperation. And when we move to the next slide, we now move to seeing the 23 targets. So like I said, the Global Biodiversity Framework comprises of 23 targets. And these are action-oriented targets, and countries need to start implementing these targets moving forward. And these targets are organized around 3 large clusters. The first one focuses on reducing threats to biodiversity, where we'll have 8 targets. And then the second group of targets focus on meeting people's needs through sustainable use and benefit sharing. And the final target focuses on the tools and solutions for implementation and mainstreaming. So these are the means of implementation, like the finance and capacity building. So when we look at the first batch of the targets, where we're talking about reducing threats to biodiversity, there are 8 of them. I don't think I want to read all of them, but as you can see, they mainly focus on ecosystems. So the first one is on planning and managing all areas to reduce biodiversity loss. We have the target 4, which focuses on halting species extinction, protecting genetic diversity, and managing human-wildlife conflicts. I think this one would relate quite a lot in line with the workshop that is currently happening. We're also looking at reducing the introduction of invasive alien species by 50%. And then, of course, minimizing impacts of climate change on biodiversity and building resilience. Then when we look to the second batch of the targets, this is on meeting people's needs through sustainable use. We have several targets there also. And the first one on managing wild species sustainably to benefit people, I think would also be very interesting in our discussions in this workshop. Also enhancing biodiversity and sustainability in agriculture, aquaculture, fisheries, and forestry. And of course, target 13, which is focusing on increasing the sharing of benefits from genetic resources, DSI, and traditional knowledge. Then if we move to the final batch of targets, these are the tools and solutions for implementation and mainstreaming. So what do we need to have in place in order to be able to implement the previous targets? So for example, we need to ensure that we integrate biodiversity in decision-making at every level. So in our workshop today, I know most of us, we know what we're talking about when we talk about why biodiversity is important, but we need to expand this conversation beyond talking to ourselves and see how we can bring in, for example, the financial actors, the private sector. We also need to bring in other sectors, that energy, for example, that have an impact on what we do in the biodiversity space. We also need to looking at target 17 to strengthen biosafety and to distribute the benefits of biotechnology. Target 20 is talking about strengthening capacity, and I think that is what we're doing in this workshop for today and the remaining days. We'll also have to look at technology transfer, as well as scientific and technical cooperation for biodiversity. And then another important target would be ensuring that the knowledge is available and accessible to guide biodiversity action. So how can the outputs of all the discussions, all the interesting presentations that are made in this workshop, how can we ensure that they're also linked to the policy and decision-making processes? And then, of course, finally, target 23 is on ensuring gender equality and having a gender-responsive approach for biodiversity action. So when we move to the next slide, now we switch around to IUCN and how does our work that we're doing globally, continentally, and even in the country, how does this link to the previous Global Biodiversity Framework? And also, how can we continue to work with different partners, including universities, to ensure that the work that we're doing informs the policy processes? So for IUCN, the vision of the organization is to have a world of living in harmony with nature, where by 2050, biodiversity is valued, conserved, restored, and wisely used. Maintaining ecosystem services, sustaining a healthy planet, and delivering benefits essential for all people. So the focus is really on ensuring that benefits for nature are also are felt by people. Then if we move to the next slide, this is just to give an example of how IUCN's work is going to be informed up to 2030, in line with the different targets, in line with the different goals of the biodiversity framework that I mentioned below. And as you can see, people are at the center of the work that IUCN does. But then apart from that, we also focus on land, we focus on water, we have different initiatives on the oceans and also on climate change. And this is guided by the need to recognize, reconnect, and restore nature. So in terms of what IUCN is doing, one is IUCN is a provider of biodiversity knowledge, tools, and standards. Some of you may have heard about the different tools, and some of you may not have heard about them, but I think they would be interesting, especially for the work that we are doing. For example, we have the IUCN Red List of Threatened Species, and I think this is, we also have the scientific commissions, where I think we really need to encourage young scientists to join the different commissions. And I'll be very happy to share the link after this with the organizers so that they can circulate, and we encourage as many African scientists to join these different commissions to ensure that science from the continent is also included in informing the global decision making processes when it comes to data from the region. But apart from that, we also have the red list of ecosystems. This is looking at which ecosystems are threatened and need their attention. We also have the green list. The green list is a tool that is used to, I don't want to say it is the opposite of the red list, but the green list is then used to acknowledge where effective management of protected areas is being done. So it's like rewarding protected areas that are doing well. And then, of course, we have the nature-based solutions and the key biodiversity areas. These are just different examples of tools and standards that IUCN has developed that can be used to implement, to restore biodiversity and reduce threats to the biodiversity. Then when we move to the next slide, so this is just an example of one of the targets. This is what target 3 is focusing. So I focused on this target because it's a target which is of interest to IUCN specifically. But then I think if you, for example, Jomo Kenyatta or other partners in the workshop can be able to look at the different targets and see which one would be of specific interest to them and how you can be able to address the different components. So this one is a very long target. It's very heavy. I don't think I want to read everything, but it's basically focusing on ensuring that by 2030, at least 30% of the terrestrial, inland water, and coastal and marine areas are conserved. And the main keywords in this target is to ensure effective conservation, effective management, connectivity, and also equitably governed. So it's really looking at how these ecosystems are governed, and at the same time focusing on protected and other effective area-based conservation measures. But at the same time, we ensure that we recognize the indigenous and traditional territories, as well as ensuring that we respect traditional knowledge. So if we move to the next slide, this is just an example of how the indicators have been developed to be able to inform, how we are going to track that. Are we meeting our target by 2030? So as you can see, we have an indicator, for example, saying that we need to ensure the coverage of protected areas. And then there are different indicators under that we can use to track and measure to see how far we are doing. So one of them, of course, is the status of key biodiversity areas. I hope you've heard about this. We also have the IUCN Green List. We have the UNESCO designated sites. We have the protected areas network metric. We have the Ramsar management effectiveness tracking tools. So it's quite a number of tools, the red list of ecosystems. So these are the different indicators that are going to be used to measure how far we've gone as a country towards achieving our target. And then if we go to the next slide. Yes, so this was actually supposed to have come a bit earlier, but I noticed after I'd sent my presentation. But when we talk about the global biodiversity framework, what countries, for example, like Kenya are doing now is to develop what we're calling the National Biodiversity Strategy Action Plans. And these national biodiversity strategy action plans are what we are going to use to implement the global biodiversity framework in the country. So the targets that you've seen in the previous slides and the specific example I've given, target 3, for example, are all included in our national targets. And the idea is to now contextualize this within Kenya to see which tools do we have available, which kind of data can we collect as a country, and how can this data then be reported to the Convention on Biological Diversity. So I think this workshop is very important and it's very timely. And I think it's very good to see the kind of data that is already being collected on the different aspects on the different targets. So for Kenya, I would say we've managed to revise our NBSAP and we have a draft. And I'm sure with time, the information is going to be shared at a wider level so that we can all see how we can participate in providing data. But apart from, thank you, I still have 5 more minutes. So apart from this, from Kenya, also other countries represented in this workshop are also involved. And I think you can find out from your respective CBD focal points how you can be involved. If we move to the next slide. Yes, so I'll just, given that I have a few minutes remaining, I'll just give examples of some of the work we are doing. So for target 3, we are working with countries and members in Namibia, Kenya, and South Africa to develop the other effective area conservation measures roadmaps. We also have some sites that have been green listed. In Kenya, we have 3 sites that are currently green listed, but we are hoping to get more. Then we are also working with the indigenous and local communities to see how we can contribute to target 3. And we also have some projects which are funded by BioPharma to improve management effectiveness and governance of protected and conserved areas. Then if we move to the next slide, this is looking at target 4. Target 4 is on invasive species. Yes, it's on halting species extinction to protect genetic diversity and manage human-wildlife conflict. So we have a commission. In Kenya, we've launched the National Species Specialist Group that brings together different expertise that are able to provide data that is used to inform the red listing process to find out the threatened species. Then we are working together with the WRTI on the National Red List of Species to update it. And then we are also having a program on reducing human-wildlife conflict. And then if we move to the next slide, we recently launched a project called BioDev 2030, which is going to be focused on Ethiopia, Kenya, and Mozambique. And the idea of this project is to contribute to the implementation of the KMGBF in 15 countries by promoting changes in production practices that reconcile biodiversity and development. The focus of this project is on target 10, 14, 15, and 16, 18, and 19. I know there are many, but I hope this presentation will be shared and we'll be able to familiarize ourselves with the different targets. But this particular project will be focusing on the policy aspects and how this can be used to inform policy reforms in the different countries. So I think I'm right on time. The final slide is just to highlight different resources that you can refer to be able to get more information about the global biodiversity framework, the different targets and actions, as well as the kind of data that is going to be required by the different countries to inform the restoration process. So thank you very much for listening to me. And I look forward to engaging further in the workshop. Thank you.
Thank you. You can hear the applause from the hall here. So still in the same session, we will listen to Professor Rasmus Heller. So Professor Rasmus Heller, sorry for, has worked on African wildlife genetic for almost 2 decades now, and he's interested in the range from practical conservation to understand theoretical evolution question. and he will give a presentation on So Professor Haler will give a presentation on African wildlife genomic conservation and phylogeography in age of genomic. Oh, is there?
Okay, thank you very much. I'm just going to see if this works. Okay. Okay, anyways, I can start by saying thank you to for the organizers for having me here. It's a great pleasure to be here, all the way from Denmark, and in particular thanks to Prof. Anne Mugai and Dr. Josiah Kuja. I don't think he's right here. He's here right now. He used to be my postdoc in Copenhagen, so I know him very well. But thanks to the ABP for inviting me here and giving me this chance to talk about our work in Copenhagen. I hope you'll find it interesting. Let's see. We cannot shift the slides. I think we're still not seeing the bottom. Maybe you need to zoom out a little bit. Yes. Okay. So I'll ask you to change the slide, maybe. Is that okay? Yes, we can have the next one. Yes. So first of all, you might wonder why we in Denmark and in Copenhagen, why we are so very interested in Africa, that we have devoted our lives, at least our professional lives, to study wildlife in Africa. Next, please. And the answer is that Africa is a very fascinating continent. If you're a zoologist, as me, so if you're interested in animals, actually, if we go back about 20,000 years back to the period of time that is known as the Pleistocene period, the whole world was full of large animals. That's what I've tried to show here in the top of this slide. So everywhere in the world was teeming with large mammal species, many of which are no longer here today, but they're still here in one place, and that's in Africa. And one of the reasons why we still have such an abundance of large and diversity of large mammals in Africa is because Africa escaped a big extinction event that happened everywhere else on the globe, roughly around 20,000 to 5,000 years ago. And I've showed here on the figure here. Let me see if I can point. No, it doesn't work. Okay, anyways, to the lower right here, you can see I've shown on the different continents, we see some bars. And here is shown proportionally how many of the large mammals went extinct on the different continents in this period from 20,000 to 5,000 years ago. And you can see the blue part is the number of, is the proportion of mammal genera that are still extant, and the red part is the proportion that went extinct during this period. So you can see in Africa, a much larger proportion of the large mammals actually survived this large extinction event. And on the the figure on the left, we've compared two relatively comparable continents, namely South America and Africa, both tropical continents of roughly the same size. And here we can see the mammalian biodiversity in the different size classes: Africa gray, South America black. And we can see in all of the size classes, Africa has much higher species richness than South America, sorry. And in particular, in the higher size classes that are to the right on this graph, Africa has much more species diversity. Okay, next slide, please. And that this fact, this persistence of large mammals in Africa makes Africa very unique globally, because it is, in the world of Huxley in 1965, it's the only remaining part of the world where we can witness this pre-human climax community of animals in its richest form. So I've chosen this picture from the wildebeest migration in the Mara to exemplify this fact, that Africa is the only place in which we can still really find this these ecosystems in which animals, large animals, they hold sway. Next, please. Yes. So this has prompted us to think and to wonder about whether Africa could be the last place where we can really study these communities of large mammals in order to understand them. What are they? How do they work? And also in order to protect them for posterity, because this is obviously something that we can all, at least in this room, agree that this is something we want to keep. This is something we want to have also for future generations. I've chosen these pictures here on this slide to exemplify also the diversity of large mammals we have in Africa. We have not only a large species diversity, so meaning many species, but also within the species, we have large morphological diversity, large ecological diversity, and all sorts of elements of diversity that we care about as biologists. For example, hartebeest here to the left, we have many, across the continent of Africa, many different types of hartebeest, subspecies of hartebeest, as we call them, giraffes as well. We have a rich morphological variety of giraffes, and these are just some examples. This is true for many, many species of African mammals. And also, as demonstrated by the picture of the lion in front of the Nairobi skyline, just from the Nairobi National Park close to us now, this is also an example of some of the threats that this immense diversity is facing right now. Being so close and having to live side by side with humans is obviously a challenge for this community. Next slide, please. So here I just want to emphasize that in this sentence, on this title of this slide, there are two elements. There are the understanding this community and protecting them. So these are the two things that I would suggest are important, or these at least are the two aspects that we are focusing on. We don't just want to protect this. Of course, we want to do that, but we also want to understand how it works, because otherwise we can't actually really protect it. Next slide, please. So the first part of that sentence is understanding. And some of the questions we are interested in when we do our research is these that I've listed here. And we use DNA sequencing. In particular, we use actually whole genome sequencing to answer these questions. So how do populations move over time? And when I say over time, I don't just mean over the last 5 years. I mean also over evolutionary timescales. How have they moved across the continent of Africa for hundreds of thousands of years? What is their natural movement patterns? Why do they move? In response to what? We also want to understand what drives the population sizes and population structure. Is it human activities? And again, I'm talking about both now, but also a retrospective perspective. So we also want to understand what has driven them in the past: human activities, climate change, other biotic interactions. We're also interested in evolutionary questions such as how populations diverge from each other and evolve into new species, because of course, we need to understand these processes if we are to fully appreciate the diversity of life we have in Africa and elsewhere. We also are interested in understanding deeper genetic functionalities, such as what are the unique adaptations that are found in each species and each population. What is their genetic basis and how did they evolve? These type of questions. Next, please. With regards to the protecting part of the sentence I put forward before, some of the questions that we can address with the type of methods we use is we can figure out which populations are genetically vulnerable. We've heard many times this conference that one of the things we want to protect is of course the species themselves, but also the genetic resources, the genetic diversity. And to know how to protect that, of course, we need to know how that diversity is distributed in the landscape and in the populations. We also can address questions such as how do we manage these gene pools that exist out there? How can we, for example, move animals around? Where can we translocate animals from and to, such that we don't, let's say, disturb too much the natural population structure that exists? And we can also use these methods in a more directly applied conservation target, such as wildlife forensics. So if we have a good representation of the genetic diversity existing in nature, of course, we can use that as reference material when we confiscate illicit items. We can figure out where was it taken from and these type of things. Okay, next, please. Yes, and a point I also want to make today that differs a little bit from at least let's say the overarching goal of the ABP, because in the ABP, at least in the first phase, the goal is to pursue an approach in which we want to sequence as many species as possible, one individual of each. But we're interested in complementing that, because we argue that to understand this and to fully appreciate this diversity, we need more than one individual from each species. Because otherwise we cannot imagine the giraffes, for example, that look so different in different parts of Africa. If we only sequence a single giraffe from one single place, we don't fully account for the diversity that the giraffes contain. It would be a very poor representation of that diversity. So our aim is to go to the population level in which we sequence from as broad a representation of the species as possible. And this is not a conflicting goal from the other one. It's a complementary goal. Next slide, please. One of the reasons why we are doing this is that we have in Copenhagen a very large tissue collection from collected more than 50 years ago, containing more than 10,000 specimens from African large mammals. They cover about 60 species of large African mammals. And we have in particular a very good representation of bovids, antelopes, these type of animals. So we have a very privileged position to address some of these questions that I've listed before that relate to the diversity found within the African wildlife species. Next, please. And this slide just shows you a representation of some of the species we're working on and that we have recently worked on to show you the taxonomic diversity. These are just examples. We have more species that we are either starting to work on or that we are planning to start new projects on. So next slide, please. We publish these results. We publish them in highly prestigious international papers. These are journals, sorry. These are just some examples from papers that we published about African mammal species in the recent years. I won't go into detail with them. So just to show you that we don't just do these analysis and then forget about them. We actually publish them and publish the data that went into producing these results. So our vision, what we want to do with our research is that we want to keep doing this. And in fact, we want to scale it out. So we sequence thousands of individuals, whole genomes. So I think it's a genomic resource that can complement very well the other things that are being generated by the Africa BioGenome Project. We want to cover at least 50 species of large African mammals. We're also very concerned about what happens afterwards. After the sequencing, of course, there's a lot of, as you all know, there's a lot of bioinformatics, there's a lot of population genetics or other type of genomic analysis. So we are very concerned about developing best practices for that. And we spend a lot of time to actually do so. Yeah, and we want at the same time also to build capacity in Africa and build partnerships here in Africa. And actually, we're running a practical workshop in connection to this conference, a 3-day workshop where we will focus on wildlife genomics. I know some of you are signed up for it. So yeah. I want to give you a few examples. I can't talk about all our research. We don't have time for that, but I want to give you a few examples from recent research that we have done. The first one I want to talk about is the wildebeest. Now, I don't know how many of you are aware, but there's actually 2 different species of wildebeest in Africa. There's the one we call the blue wildebeest. It's one to the left, lower image here. That's the one you know from Masai Mara, Serengeti, all of these other places. That's the one that is really super abundant and widespread in Africa. We can see its distribution over there on the map in the blue color. And then we have its sister species, the black wildebeest, on the right here. And on the distribution map, there you can see its distribution is colored in black over there. It has a much smaller distribution in southern Africa. Actually, the black wildebeest used to be as numerous as the blue wildebeest, but due to persecution from humans, it was hunted almost to extinction in the 1800s, with just a few dozens of animals being left. But through a determined conservation effort, this species is actually now doing much better. But these are the 2 species we wanted to analyze. And yeah, next slide, please. Sorry. And some of the results you can see here, I don't know how well you can see because the image is not too large, but in the top you can see the map of with the dots representing where we have samples from. So you can see we have samples from most of the distribution range of these 2 species. And yes, and I want to just quickly go through the main results on the lower part. If you zoom out a little bit again, yes, we can see principal component analysis, PCA, which shows which visualizes the population structure in these animals. And we can see over here that, I can maybe point here, we have the black wildebeest individuals over here on this plot, and we have all of the blue individuals then on this line here, over here. So we can visualize the genetic structure within the species with these type of analysis. And if we look at something else to the 2 plots on the right, we estimated or measured the genetic diversity in the different individuals. And here we can see, if we look at the upper plot, we have made a box plot of the genetic diversity in the different populations. And you can see here it looks a little bit messy, meaning that populations have a wide range of different levels of genetic diversity. But if we move to the lower plot, here we have done something in which we take recent inbreeding out of the genetic diversity estimation. This is something that can be done analytically. And when we do that, we get a much clearer image in the lower right plot, yes, where we can see that the populations to the left, which are the southern populations of blue wildebeest, they have higher genetic diversity, and the populations to the right, which are the Eastern African wildebeest populations, have a much lower genetic diversity. Okay, next slide, please. We also wanted to investigate something else because this dramatic imbalance in the abundance of these 2 species, it has been feared that there would be genetic swamping of the black wildebeest from the much more abundant blue wildebeest. If you remember, I said that the black wildebeest had been reduced to very low numbers. So there's a risk that actually, because these species can interbreed, they can hybridize. There's a risk and a concern that the blue wildebeest has genetically swamped the black wildebeest by its introducing its genes through gene flow. So we wanted to investigate this, but actually we surprisingly found that exactly the opposite has taken place, that in the southernmost populations of the blue wildebeest, there is some black wildebeest genetic material. So there has been integration, but it's in the opposite direction of what everyone has been fearing for a long time in the conservation circles. So that's a surprising and also in some respects, maybe a positive finding. So we should not be so concerned about the black wildebeest because it has actually not been genetically swamped by the blue wildebeest. It's the other way around. Next slide, please. So here is just a demonstration of what the whole genome data can actually give us. Here we look in two different individuals of a blue wildebeest. One is from the southern African part of the range. The other is from the eastern African range. And here we noted across all of the chromosomes in the wildebeest, in which parts of the genome do the individuals contain some elements that actually came from the black wildebeest. So introgression, which part of the genome has introgression from the black wildebeest. And here we can see, we can see or we can corroborate that the southernmost blue wildebeest populations have many places in the genome where they have black wildebeest genes, whereas the ones in East Africa have barely anywhere. Actually, they have nowhere. Those few purple lines you will see in the right-hand plot are what we can call noise, analytical noise. They're mistakes. So the left-hand individuals are southern, the right-hand individuals in Eastern African blue wildebeest. We can see where in the genome it is actually black wildebeest. Next slide, please. We can also compare the two species genome-wide and figure out where are they most genetically differentiated. And this is something that can help us understand why are they even two different species? What makes them different? Which part of the genomes make them different? And if we do that, this is what is done here in this plot on the top. We can see we find one particular region where differentiation is highly elevated. You can see these dots going up in this peak. And if we look at what genes are positioned in here, there's some interesting genes. There's a gene called OCA2 and HERC2, which are both linked to color morphology or coat color in other species. So maybe this could be the reason why they actually look different, is because they're differentiated in this particular region. We also find some hemoglobin genes in this region, which could account for or could be related to the fact that they have very different movement ecology. With the blue wildebeest, we have very migratory species, and the black wildebeest are less so. Next slide, please. Yes. So the final thing I want to say about wildebeest is that we also did something else, because as I said before, the blue wildebeest is known for being migratory. It's essentially it's made to migrate. Everything about its biology is suited to migration. So when we compared the blue wildebeest populations that still have intact migrations, for example, the one in Serengeti and Mara, that I'm sure you all know, to the populations that used to migrate but can no longer migrate because humans prevented them to do so, we saw something very interesting. And we saw that both in the Eastern African part of the distribution and also in the Southern African. So here we see on the right hand here, we compare those populations that are migratory. Those are the ones that have the, you can see maybe on the graph here, the red ones on top. Those are the migratory populations. And as we move down this line, you see the non-migratory populations. And what is depicted here is genetic diversity and inbreeding. So what this graph tells us is that both in Eastern and in Southern Africa, the migratory populations have higher genetic diversity and lower inbreeding than the populations that can no longer migrate. So this is a very tangible evidence that the populations that are being prevented from migrating, which is their natural ecology and behavior, they have compromised genetic health. So this is quite interesting from a conservation perspective, I think. Next slide, please. The other case I want to give, I don't know how I am on time, but I just want to maybe go quickly through this, is from the recent study we did on giraffes. And the interesting thing about giraffes is that it has recently been proposed that there is actually not just one giraffe species, there's many. And the reason why this has been proposed is that I've already given the example many times before that the giraffe really has a wide morphological, wide diversity in morphology. And people have been doing genetic analysis that prompted them to conclude that it is not one species, but 4 species. We've been a little bit hesitant to believe that result, so we wanted to do our own analysis. Next slide, please. And what we did is, again, sequence whole genomes from most of Africa and combine them with those already published. And what we first did is the same as before. We wanted to estimate the population structure, continental Africa scale. And we do actually confirm that there are 4 very different groups. But sort of embedded in these 4 groups, there's a number of further subdivisions of the giraffe. So it's a simplification to say that the giraffe is 4 different things. You might as well argue that it's 10 different things or 12 different things or some other number. So the 4 species is a little bit, I would say, tentative based on this. Next slide, please. We also found something else which was quite interesting and which goes to the question of whether there's 1 species or 4 species. We analyzed gene flow between the different types or evolutionary lineages. And what we found, to our great surprise, is that there's a lot of historical gene flow between these different lineages. Even though they're very different, genetically differentiated, we can find evidence of past gene flow, very clear evidence. And this graph here on the left summarizes how much gene flow has been taking place between these lineages. And the most conspicuous thing that you see, I don't know if you can read the numbers well, but the reticulated giraffe, which is right here in the graph, is a hybrid, 60% coming from a northern giraffe lineage and 40% coming from a southern giraffe lineage. So the reticulated giraffe, which is found among other places in northern Kenya, it's the red dots over there on the map, is essentially a hybrid. You can call it a hybrid lineage, which was never known before. So obviously an interesting observation, but also interesting because it tells us, okay, if these very differentiated giraffe lineages, if they can have gene flow and hybridize with each other, and they have done so naturally in the past, are they really different species or not? I'm not going to make that judgment. I will just put it to you that this questions very much whether we can talk about different giraffe species or not. Next slide, please. Another thing that we can do, these 2 cases that I just mentioned were very species specific, wildebeest, giraffe. Now, when we start generating data across many species, these population level data sets, we can start comparing them. And we can start asking interesting questions such as, where do the different species have barriers, genetic barriers? So this is a method here that actually visualizes that. You can see this is the continent of Africa. And overlaying on top of that is a genetic analysis that show where is the genetic discontinuity in these species. That's marked by the red or brown areas on each map. Now we compare 6 different species of African mammals, and we can start addressing, do they have any shared barriers? Do they have any barriers that all species have? Do they have some barriers that are unique to some species? And those that are unique, are they then shared by a subset of species that perhaps share a particular ecology and things like that? So this is just an example. Of course, this only really becomes interesting when you start having many, many species. So you can start understanding what is it really that governs the movement of large mammals in Africa as a community, not single species. And again, when I'm talking about movement, I'm not talking about movement over the last 5 years or 10 years. I'm talking about movement over evolutionary timescales. Oh, actually, both evolutionary timescales, also recent timescales. So both of them. Yes, I'm almost done now. I just want to finish by saying that the work we are doing, of course, requires African partners, and we already have many. But the reason why we have actually entered into a dialogue and become associated with the African BioGenome Project is exactly for this reason. We want to get in touch with African research communities because we want to do it together with African research communities. This is both good for us, it's good for Africa, and it's really the right thing to do. So this is something we are very, very concerned about, and I just want to put it out there that as an open invitation to always get in touch with us if you're interested in collaborating on some of these questions, and building new projects, applying for funding together, these kind of things. And also, I should say, we also have a big emphasis on training, building capacity in Africa. We're doing it. We've been running a workshop last year in Rwanda in connection with the ICCP. We're running a workshop and a training course this year, and we hope to keep doing this more and more as we build this infrastructure and collaborations that are necessary to do that. And of course, try to get funding to do it. Next slide, please. So these are just some examples of some of our good partners that we already have and had for a long time. Some of them, we have some very good partners in Uganda, where we have helped to train dozens of PhD students over the years. We're also helping them to build a computational infrastructure and biobanking infrastructure in Uganda. And then we have recently entered a dialogue conversation with the Wildlife Research and Training Institute here in Kenya that we really hope to also build a strong collaboration with here. So these are just examples of some of the stuff we're doing in collaborations we're trying to build. Next, please. And this is just the final slide to acknowledge some people. I, in particular, want to acknowledge the people in Copenhagen that are not here, but that are behind much of this, and some of our best and longest serving African collaborators, especially Joseph Ogutu, who some of you might know, who has helped us a lot and been part of our work. Sami Wambua, who's helping us do the workshop in Kilifi in the coming days. Yeah, and I just have one final slide, is just a reminder that this workshop is taking place in Kilifi, and yeah, hope to see some of you there. Yes, thank you very much.
Thank you, Prof. Let's remind that Prof is coming all the way, as he mentioned, from Copenhagen to give a presentation, and secondly, we appreciate for that. We will move to the question and answer, the Q&A session. Yeah, for those who have questions, you can raise your hand and we will give you the mic. And also online.
Thank you very much. I'll ask Professor Hela. You talked of there having been a bit of admixture, and I think it's a move from the black to the blue. I don't know. I'm interested to know maybe when that happened. Yeah, because we've got humans also moving during that time. Then equally, the last question, you talked of there having been an extinction event which didn't have such a big impact on Africa. Maybe why was Africa not affected largely? Thank you.
Thank you. Okay, my name is Derek Oma from Kenyatta University. Yeah, and my question actually is directed to, what is the name actually, sir? Yes. Okay, thank you. So in actually tree planting actually project that I've seen in Kenya, yeah, and I would categorize it as, you know, I'm looking at Kenya, and then within Kenya, I look at urban centers and upcountry, yeah. So I'm gonna focus in urban centers, and then let me narrow down to Nairobi, and then to Eastlands, Nairobi. Yeah, there's actually a huge problem with the, you know, environmental conservation in Eastlands, yeah. So what actually efforts are being taken, and I'm referring specifically to Nairobi River, which is actually heavily polluted with heavy metals, yeah. So what actually I'm kind of wondering, what actually the efforts in conserving, environment through tree planting and maybe targeting, the river to provide, water for the seedlings. So maybe within the process, where actually are the efforts and what is actually being done roughly with regards to that? Thank you.
Okay, so my question is, my name is Kisoy, and my question goes to Andrew. And speaking of the species of plants that you intend to, of course, you've identified and you told us that it's 58,000, roughly there. So did you focus, or your institution focused on identifying some of the areas or regions in Kenya? Because we keep on hearing about tree planting since time immemorial. And part of Kenya is usually desert or semi-desert. And where I come from, I haven't seen any effort by the government or any institutions that partake the tree planting. So I don't know whether the species you are focusing on are climatic specific areas like the highlands and so forth or not. And the second question goes to Prof. I don't know what some of the factors that facilitate the gene flow among the giraffes, especially in northern Kenya. Thank you.
Okay, thank you for the opportunity. I'm Dennis James from JKU, and I have a question to Prof. Given that we have been facing extinction of various species, mostly in the wildlife sector, I'm trying to think of the possibility, maybe, is there a possibility of using science, and right now we are talking about biotechnology, to curb these, to prevent some species from going extinct. For example, through the gene, through modifying the genes of various species, and then trying to do a duplication of various species. Of course, I know it can have some ethical issues, but can we go to that direction to prevent some species from going extinct? Thank you.
So let me read out the questions that are being posed online. Dr. Andrew, please comment on the natural regeneration as opposed to tree planting as a working strategy towards restoration and increases in biodiversity. The next question is for Ms. Catherine Mungai. A good presentation. Can you give us an explanation on how you can ensure communities are shared biodiversity benefits fairly as per your goal 3? Secondly, you never touched on microbial biodiversity. Is it not important to conserve microbes and get benefits of 99% of useful microbial products like foods, medicine, pharmaceutical, climate regulation, etc.? There's another question for Professor Heller. Is there any particular reason why the black wildebeest is tenacious? The reason for the tenacity of the genome of the black wildebeest? I think those are the ones that I can see.
Thank you. You can answer. We will maybe go on this for the second round of the question if need be.
Nice, thanks. You want to go first, Professor?
At least I'm struggling already to remember the question. I saw you were noting down. You are smarter than me. Okay. Okay, I'll try to take them in order if I can remember them. I think there were two very good questions from here. The first one was about when was the gene flow between the black and the blue wildebeest? We actually can try to we try to estimate this, and our estimate is several thousand years ago. So it's not a very recent thing. It's something that took place, we can say, naturally, at least before humans started playing a big role in interfering with the distribution of these species. I should also say, I didn't have time to say that very clearly, that where we found this integration is only in, I think I said it, but briefly, is only in the southern blue wildebeest, not in the eastern one. And that's, of course, for good reason, because this is where they came in contact with the black wildebeest. The black wildebeest were in South Africa. And then what we believe happened is that the blue wildebeest probably expanded its range at some point in time. And this is the time at which they then got into contact and the gene flow started happening. This is our hypothesis, at least. Of course, we cannot ultimately prove this. But several thousand years ago, I don't remember exactly the right number. I think on the order of 6,000 to 10,000 years ago is our estimate. With regards to the extinction, Pleistocene extinction event, and why did it not take place in Africa? That's a great question and very hard to answer, because nobody knows, I think. There's a lively debate about this, has been for decades in the scientific community. And I don't think anyone has the answer, but there are some hypotheses that goes to, so as you probably know, humans actually originate from Africa. So essentially humans everywhere else except in Africa are, you can call it, an invasive species. And maybe because the wildlife didn't co-evolve with humans on the other continents, that's not my hypothesis. It's a hypothesis that had been put forward. Maybe because wildlife, so to speak, was not accustomed to humans on the other continents. And then when humans suddenly came, they were too, what's that word? Sensitive, or they couldn't cope with the pressures that came with human presence, be it hunting or starting fires and whatever humans have been doing. So that's one hypothesis. I don't know if it's true or not. And then I'll move to the next question, which was, I think, from down there, the gentleman down there, that was about gene flow in giraffes, if I remember, in reticulated or northern. So why it is taking place, I think was the question. I have no idea, I have to admit. Also, this gene flow is something that happened even longer ago than the one between blue and black wildebeest. It happened probably hundreds of thousands of years ago. So why it took place, I don't know. One hypothesis is that's one hypothesis that we put forward in this paper. So how can the giraffes at the same time be highly differentiated, yet we can document that they have gene flow between them? One hypothesis is that giraffes are very responsive to climate changes, so they have been periodically isolated by climate change, then becoming highly differentiated, but at other times brought back into contact with each other, and therefore having gene flow simply determined by the environment and not by anything else. That's I think, at least my best hypothesis for that. Then the next question was more about this, actually some of the things we heard yesterday about de-extinction, I think, by using technology. This is something I'm not an expert on, but I can give you my, I mean, in theory, we are probably approaching a point in which it is technologically possible, but I would probably say that's not the main concern right now. The main concern, if you cannot even keep the species as they are now, I don't really see how just cloning them and putting them out again would solve anything. I think we need to face the problems that they're facing now before we start repopulating or rewilding. That's my own personal opinion. And then I think there was one online question, and it was about tenacity of yeah. So if I understand that question correct, yeah. So why is it the black wildebeest genome that prevails, I guess, is the interpretation of the question. So actually, I don't think it has anything to do with tenacity in that sense. I think it's just a consequence of how the gene flow occurred. So as the as the blue wildebeest expanded into the range where the black wildebeest was already there, our hypothesis is that by through interbreeding every now and then, the black wildebeest simply left a little remnant of its genome in those southern blue wildebeest populations. So it's simply a consequence of the geographical nature of this contact and not anything else. But I don't know, this is by speculation on my part. I think that's it. And then...
Yeah, thank you so much. So yeah, I have 3 questions here. The first one from the gentleman there. I didn't get your name. It's about green space restoration in Nairobi and the challenges you're facing, particularly with or around Nairobi River. And I think this is a contentious issue because it has been going on for some time, and that's why you're asking about this. And I am aware of a few projects that are going on in Nairobi, and just one that is going on in around Nairobi River. But the challenges you mentioned about water quality and having to use that in I think in restoration or using that in tree nurseries, it's a challenge. And one of the quickest solution would have been to, it's the reason I said it's contentious, it's to move people away from the riparian land, because this is again, another, how the major, just eliminating threats as the first activity or the first option, and reducing the pollutants and pollution that is going into the river. And next is coming up with identifying reference sites within the river, probably in the upstream, identifying species that you can use for restoring riparian or taking care of the riparian land. Then that will solve the problem of water pollution, and probably in the long term you'll have clean water in Nairobi. I think some of these projects are focusing in addressing this problem using these approaches, including the government. And the next question from Kisoi, this was about species threats and planting trees in, or understanding that Kenya has diverse climatic conditions. 80% of the country is arid, actually. And when I'm sure maybe your question is to connect to the large number of tree planting initiatives, 15 billion trees, where are these trees going to be planted? And again, it's the same question that we're trying to address now, having to identify tree species and getting the right tree for the right places, and assessing projects that are doing this restoration work, promoting and recognizing best practices. Those tree planting projects that are happening in the right area, addressing problems and not even causing more problems. In the manual that I presented here, the manual that we are using to assess these projects, criteria number 1 is looking at protecting existing biodiversity. And this addresses that problem of going back in time using different tools like remote sensing to go back in time and understand how these landscapes used to be years ago. And and then identifying and developing reference models to restore these ecosystems in a way that it doesn't replace what used to be there years ago. So in the savannah, for example, where tree planting activities are going on, the main problem is that there's not a quick balance, we are not maintaining that natural balance between grasslands and woodlands. And in the end, we end up having replacing or displacing biodiversity, that native biodiversity, with other biodiverse ecosystems that are also negatively affecting the larger landscape. So it's captured in that biodiversity manual, and it's a link that I can share so that you understand the best practices that are being employed. And the third one from an online participant, this was on, they were asking for me to comment on natural regeneration as opposed to tree planting. Again, tree planting has been fronted, and I mentioned this, I think, in the talk, as the go-to solution because it's quick fix. And you find that even most of the funding organizations, they need numbers, they want to see trees that have been planted, but they're not willing to support that research aspect or any other aspect that is not directly linked to, that is indirectly linked to to restoration. So with this condition in the funding landscape, you find that most of the implementing organizations, some of them are CBOs, and they don't have, they lack that connectivity between science, or that connection between science and practice. So you find that most of them are going straight to the quickest option, which is tree planting. In essence, if you give, it's a whole ecosystem restoration, it's a whole continuum. And if you give nature a chance, then it can easily bounce back with the right support. But then it needs a lot of research in understanding the level of degradation, identifying threats that a specific ecosystem is facing. And then with all this data in place, then you can make informed decision whether tree planting is the go-to solution, or you can support the ecosystem to naturally bounce back.
Thanks. Thank you for your answers. I'm not sure if there is any second round since we are running beyond schedule. This Catherine online, whom has a question from an online participant. Okay, maybe she's not online. I think we will stop here, and thank you again for your presentation. Please let's give them applause. Thank you. And we'll go for a short tea break of 30 minutes. And please take time to visit the stand of the partners outside. And for the online participant, we'll resume in 30 minutes. Thank you.
Let's go straight to life sciences solutions. Okay. Life sciences group category page. When you come to this page, you get to see key divisions under life sciences group, and these are biosciences division, there's clinical science division, and there's genetic science division. Okay, so I'll just go straight to division, and from here I'm able to see some of the products under biosciences divisions, and quickly I'll just go straight to just one items. So I'll click here. Let me just choose any of the items around. So I can just click product. It takes me now to the product page for fetal bovine serum, FBS. And from here, as a client, if I'm interested in the product, I'm just able to decide and just request for price and availability in this website. Okay, so to make an order or even request for price, you just click on this button here, which takes you to the checkout section. And from here, this is where you're being asked a few questions before the order is actually placed. Yeah, so when you're here as a client, you just get to fill in a few information. So I'll just do it. Probably I'll just say I'm from ABC, then just email. We are from ABC company. There I'll just put in my name. So be John. My name is John, and my name is Awad. John Awad. Here I can either put in the phone number. I think the phone number is actually optional, but also good so that the sales to get to and in a manner possible. Okay, yeah, so I'll just put in a random number. Yeah, that is fine. And as you can see down here, we have the exact product that we're actually inquiring for. And the next thing is just to order as the client. So I'll just click here. The order is being processed, and shortly the process should actually be successful. So as you can see, I'll just get this message: thank you, your order has been received, and it will go now to the relevant department, the sales department. They can now check with the client to advise exactly how much the costings and everything. Yeah, so that's it for our presentation. I hope everything was helpful.
Santangezin Mapori, a Deputy Executive Dean at the College of Agriculture and Environmental Sciences at the University of South Africa. So this is very excellent news for us and a good opportunity for us seeing the PacBio platform actually growing in South Africa, especially now the acquisition of PacBio. Thank you.
My name is Matati, an NGS lab technician here at Ngaba Biotech. Using the new review system, fewer kits are required in library prep up to sequencing across the various applications we provide. Using the new streamlined workflow, library prep is quick and efficient.
My name is Hamilton, and I'm part of the NGS team at Ngaba Biotech. And one of the stuff that has data that I've looked into, I think one of the advantages of the system is that not only does it produce long, high quality reads, but when you actually work with these reads, it gives you one of the most comprehensive views of the genome. From the review data, not only do we have access to the genomic landscape of the human genome, but we also can zoom down into single nucleotide variations, which is very important for right applications. So it makes sense the data to come off this instrument and for its future applications.
In addition to this, which is our long-way platform, I'm thrilled to introduce to you the new PacBio system, which is a short-read platform. The ONSO system is based on the sequencing by binding chemistry, which is a major contributor to the type of data that you get. By the type of data, I mean the Q40 plus level of accuracy. The independent flow cell lanes and the external clustering brings a level of flexibility that has never been seen in the short-read market before. Accuracy will allow our customers to sequence at much reduced depth and also reduce the cost of sequencing. We are so thrilled to have this system offering services to our customers.
Video, I'm gonna take you through the whole process of ordering a product from the same website. So to start off, Africa Biosystems as a company focuses on key sectors, right? So for us to explore these key sectors, you just put your cursor just directly on the navigation bar. Put your cursor. And you can see here, we have 7 key areas that Africa Biosystems actually concentrates on. And these are life science solutions, there's analytical solutions, there's forensic solutions, diagnostics, service and support, and lastly, we have general products. In either of them, we get to find more categories inside, okay? So in today's demonstration, let's go straight to life sciences solutions, okay? And actually here on the life sciences group category page, when you come to this page, you get to see 3 key divisions under life sciences group. And these are biosciences division, there's clinical science division, and there's genetic science division, okay? So I'll just straight to biosciences division and just click on that. From here, I'm able to see some of the products under biosciences divisions, and quickly I'll just go straight to just click one of the items. So I'll click here. Let me just choose any of the items randomly. So I can just come here and click this fed bovine product. It takes me now to the page for fetal bovine serum. Yes. And from here, as a client, if I'm interested in this product, I'm just able to decide and just request for price and available.
So, welcome back, everybody. And this session is for the young scientist, AfricaBP young scientist. And young scientists are key contributors to the success of African BioGenome Project. As you have noticed, recently, we even have fellowship for the young scientists. And with the young scientists, we trust that the future of Africa and genomics is well taken care of. So today, we have got 3 presenters as young scientists who are really working on interesting projects that will listen to and ask questions. So the first presenter is Vincent. So the first presenter is Vincent Habimana. And Vincent is a PhD scholar working on animal genetics at the Sokoine University of Agriculture in Tanzania. Vincent holds a master's degree in animal genetics from Sokoine University. He also has a bachelor's degree in veterinary medicine from the University of Rwanda. He is conducting his research in collaboration with the Succeeds Africa Center of Excellence for Infectious Diseases at Sokoine. As well, he also collaborates with IRI. Topic for today is genetic variation of HSF1 and HSPA6 genes responsible for heat tolerance in dairy cattle breeds reared in Tanga region in Tanzania. And here we have Vincent. Take over the platform. Let's welcome Vincent.
Thank you so much for the introduction, and thank you so much for the opportunity to share the preliminary findings of my work. As the speaker said, I'm Vincent Chabman. I'm registered as PhD scholar at Sokoine University of Agriculture. I'm here to share the work on genetic variation
in partnership with 10KP project, and 10KP project is that project more or less similar to what we do. Now with PacBio, GP is another project whose focus on sequencing the vertebrate, and then their goal is to and we partner with them, and with them using the Inkaba platform, because Inkaba is where the technology is, where the sequencing happen, their sequencing center. So with UNISA, we sequence around 10 species. And then with VGP, we partner and sequence these three. We have here two bats, and these species here look like snakes, but snake, that's the name here. And with the South Africa able to sequence the these species here called Hill. I want to pronounce that name because this is sequenced on the continent. Before that, so this is among the first plant species sequence, alongside these two other one where we sequence in partnership with UNISA. So the technology used, we do also run what we call the fellowship program in the subcommittees, and some of the beneficiary of the program was one who was the one to present before me, but I think he will present. And in the ITER, with the ITER in Nigeria, we had 10 fellows, and their interest was on these plant species called in Nigeria a language Akbalumo. Okay, and in English is just called the African star apple. So, and with the AGC in Morocco, we had 10 fellows as well, and it was still a plant species that was of interest, the Vachelia gumifera. Yeah. The different technology used, different technology have been used during the sequencing. AGC and AfricaBP fellowship program in Morocco. Illumina was the one that we use and we generate the short read sequence. And with ETR and AfricaBP, in Nigeria, we use the PacBio in combination with ONSO to generate the short and the long read sequence. And with Inkaba challenge, there is also Inkaba challenge. Inkaba, I think every year run a challenge project, and they will sponsor the project in terms of sequence. implemented with it, but so we might move at the moment while we expect to get co-funding on the maybe sometime the requirement from the partner, how we what species they're interested on, and if it's African species is also an interest, so we go for it. Yeah, that's what I would say. But we are very interested on plant species as well.
All right. Thank you for answering that. Any other question? All right. Please.
Thank you for the presentation. My name is Beatrice Nyaga. Mine is not a question, it's a request to Vincent. If you've published your work, kindly share with the others.
Okay, thank you, Beatrice. Normally the work is still under review from the co-authors. So far, the first preliminary comment I received, the results are with minimum acceptance, but they have to work, address, go deep, and that is, I'm okay with that, to do the discussion. And I expect after later on, I will share with Dr. Steve, he's here in the room, and then in November, I will share. So with that in mind, I'm sure when the work is published, maybe next year, the IBR here research team will find it, and so you can find. Thank you.
All right, thank you very much. Any other questions? Please pass this for me. Just to remind Mr. Frederick Kebaso to be ready to present next when we finish taking the questions.
Hi, this is for Yves. Just out of curiosity, I was just expecting from your presentation, maybe you show the data management plan in your capacity. So maybe share a link where we can learn from the, because obviously there's a lot of data that's going to be produced. So this is just out of curiosity.
Yeah, thank you. Yeah, when there's data, always there is sometimes conflict of interest. So, and also sharing some information can also be conflict of interest. You have to be okay with partners. Can this be shared or not? Can the information be held? So, yeah, I agree with you. And the data management is actually, we have, as you saw, the different subcommittees. We have actually subcommittees that deal with that aspect, the drafting MOUs with partner, which is partnership where Dusha is member. So that's as for us in the sequencing and assembly subcommittee, we won't deal much with data accessibility. That's not our role. Our role is the data accessible? Yes, we do the job that needs to be done. Thank you.
All right, thank you. So we'll go ahead and have a presentation from another young scientist, Mr. Frederick Kebaso. I think he was introduced, but for the sake of the audience online who might not have had his introduction. So Frederick is currently a bioinformatician at ICIPE, and he has a BSc in molecular and cellular biology from Kenyatta University, and is currently undertaking an MSc in bioinformatics, and in particular application of bioinformatics in resolving issues around cancer. So today he will be making a presentation to us around his current research work. And Mr. Frederick, if you're there, please take it away. Thank you.
Okay, thank you. So, and you confirm you can hear me?
Yes, we can hear you.
Okay, thank you, and sorry for the internet issues that you're having from your side. So I am going to talk about my MSc work on a de novo genome assembly of the camel kid and annotation of chemosensory genes. And as had already been mentioned, I was one of the beneficiaries of the first fellowship to Morocco. And there we were extracting DNA and assembling the genome for the Gumifera plant. And it's simply an acacia, one of the acacia species that is surviving in the deserts. But for today, I'll be talking about the genome of an insect. Okay, so as I have already been introduced, we'll go to the next slide. So the Hiboboscus camelina, or the camel kid, is one of the kids that invests the camels. And I'll be concentrating on the chemosensory genes of these insects, as you can see on the image on your right side. Then there is an expanded or a scanned image on the right side. And from there, we're able to see that the camel kid has an head, but it has no extended antenna. Next slide. Okay, so on the background, Iboboscus camelina, or the camel kid, transmits many pathogens, including the Candidatus anaplasma cameli. But on top of that, there are also other effects that it has on the camels. It has painful bites. It has alopecia. Alopecia is the loss of hair on the skin. There are also skin lesions. We also have the weight loss, and also it causes anemia due to a large amount of blood that they take. For example, here in a study previously, it shows that a single camel kid requires about 1.5 to 4.5 milligrams of blood in a span of 6 hours. So, and they feed in 3 to 13 minutes. So you could expect that when you are having a lot of investigation on a single camel kid, there will be large loss of blood and it can lead to anemia. Let's go to the next slide. Let's go to the next slide. Can't see the slides moving from my side. We can go to the next slide. Seems the slides are stuck. I can't see them moving. Maybe you can reshare them again.
Months of protests.
Frederick, can you hear?
Okay, I can hear you. Okay. I
Okay,
wanted to
you can
try and
continue.
hear them from the side, but okay. Now, on the background, the camelcade or the bobo seeds, because all of them, they belong to the Ibosidae family, so we can refer to them as Ibobosids. There are 7 known species of these, and they have developed a robust chemosensory system that has aided in the successful ecological establishment within their respective niches. So most of the insects, they use the chemosensory system to process their cues, and the chemosensory system is responsible for olfactory signaling. So the chemosensory genes, they enable the insects to find their blood meals, their mates, and also to evade from their predators, and also to find their larvae position sites and to detect predators. Can you go to the next slide? Or maybe, can I try sharing the slides from my side? Because I can't see that, whether they're moving. Maybe you can stop sharing, then I can share them from my side and see whether it can be possible to move them. Okay, so I hope that is okay. My slides can move, eh?
Yeah, they can.
Okay, thank you. So the objectives of this study was to first do a genome assembly, a whole genome assembly for the camelcade. Then after that, annotate the genome, then now focus on the chemosensory genes of the camelcade. Now, the type of data that we had both long reads from Nanopore and also short reads from Illumina. So, and this is the sizes of the data that we had. For the long reads, we had both the male and the female kids, both single individuals, of course, as expected, both single individuals. Then the size of the female was 12 GB and the male, the size of the female was 12 GB and the male in the GC content. The size. Now, to assemble the genome, annotate and identify the chemosensory genes. This is now the whole workflow that this study followed. Step 1 was to do read cleaning and filtering to remove the adapters, the contaminated reads, and also to drop any short reads, or also to drop reads that may have accumulated errors from the sequencing step. Then now we proceeded to do the genome assembly and quality assessment. In genome assembly, I used Fly. Fly is one of the long read assemblers that we have around that performs good. And again, I can mention on this, I tried also other assemblers, but Fly performed better. And I'll explain what I looked at when I see Fly performed better or give me a good assembly. Then I used Pylon to improve my assembly or to correct my assembly using the short reads from Illumina. Then because this was a de novo assembly that I was performing, meaning I didn't have a reference, I used the repeat modeler to build a library or to model a library of repeats from de novo, then use the built library now to mask the repeats in my genome using repeat masker. Then after that was to assess, of course, the quality of my genome. In this case, I used ComplereSum. Sometimes you can use Busco or Quast, but I prefer ComplereSum because of its accuracy. Then I proceeded now to do genome annotation. And in the genome annotation, I employed Breaker, which did the structural annotation of my genome. So it provided me with a GTF file, of course, with the gene boundaries, shows you where the gene starts, the start and the stop. Then it gives you also the coding sequences. Then from there, I proceeded to do functional annotation. In this case, I employed the AutoFinder to build clusters to cluster the annotated genes into various gene families. And then from there, I identified the chemosensory orthogroups. Now, but having the chemosensory genes, there are other steps that you need to confirm that these chemosensory genes are functional and they are actually they are available in the genome and they are active. Number one was to ensure that my chemosensory genes, do they have the functional domains? So I did a functional domain search against the NCBI-CDD domain search. Then I did amino acid alignment. Here, the aim was to confirm, do we have the correct residues at the correct positions to enable this gene function well or bind to the odorants in this case. Then also I did reverse blasting and manual annotation. So in the preceding steps, I explaining all these results that came from this workflow. Number one was from genome assembly. So in genome assembly, remember I said I used Flyer and also there are other assemblers that I employed. So these are the results from Flyer. The genome size was 132 for the Camelina female, and for the male, it was 130. And also I looked at the contig N50. This shows the contiguity of the genome, how continuous your genome is. So the male genome had 1 MB, then the male genome had 419 KB. And also you now look at the number of contigs, and of course you could expect this with a contig N50 of 1 MB, you could expect this, and also for this one, you could expect to have a higher number of contigs. Then this was the completeness of my genome. And this completeness is at the dipteran level or using the diptera lineage. The Camelina female was 95% complete and the male was 94% complete. And all these are the other statistics that you can look at when you're checking the quality of your assembled genome. I'll go to the next step, which was now after annotating your genome and extracting the chemosensory genes. When checking the chemosensory functional domain, for this case, I'll be using the odorant receptors. The functional domain here is the 7 transmembrane domain. And these are some of the genes that I found out. I will give a table at last showing the total number of genes that I confirmed to be functional. So here, HCS is H. camelina male. These are some of the genes that I found out and I confirmed to be functional. Then when doing the blasting, of course, against the NCBI-CDD, I did use the available, already known well models that have been characterized to do that. This is from Glossina fucipes. It also contains the same. This is an odorant receptor from Glossina fucipes, which also contains the 7 transmembrane domain. And this is the novel, or we call them putative odorant receptors from H. camelina. This is from Glossina breviparipis again. So the coloring here just shows this is a model that is in the database, so it is more specific. That is why it is already found here. It is a specific match to the database. And also this is for Drosophila, etc. So these were models that I was using also to match with mine. Then now when we go to the looking at the conserved amino acids, in this case, I'm using another gene family, chemosensory gene family. In this case, the odorant binding proteins. And one of the features is that they have a 616 signature. And you can see there is, when you do the alignment, this is what you expect. You expect to see 616s that are matched. So here I'm showing from C1 to C6. And the alignment also includes other model or well-characterized odorant binding proteins. It's one of the chemosensory genes of the insects. Then now on manual annotation, this is where now we are looking at the gene. Does it contain the expected nucleotides and the expected boundaries, so that when splicing is occurring, it occurs normally as expected. So this is just a feature. I'll show the real gene that I found and how I'm looking at this.
Frederick, please kindly summarize in the next 3 minutes.
Yes, I'm almost finishing. Okay, so this is, I was looking at the start codon, which we expect to be ATG. Then we are having also the exon and intron boundaries, where we expect the GT and AG, then the stop codons to be at the end. Then this is now one of the genes that I found, an odorant binding protein. And in this case, I was using the Artemis software, which I was able to visualize the exons or the coding sequences, the introns, the start and the stop codons. In case there is any mismatch, it also gives you a mechanism on how you can tweak that. Then this is now the final gene counts for both the male and the female. And also I'm comparing that to the closest species, not a reference in this case, but the closest species, because when it's a de novo study, you can't do it in isolation, but you have to compare it with the closest species that is available. So in conclusion, we see that the genome size for the Bobosca camelina is reduced, and we associate this to the 50% of the repeats that we are seeing that have reduced when you compare them to the Glossina morsitans. And also the chemosensory system genes are reduced, so we see them from this other side. So I could like to acknowledge everyone on this slide, and thank you.
All right, thank you very much. We'll have one more presentation before. What do we have questions for? We can take 2 questions for Frederick because he's online and the internet is favoring us right now. And then we can go on to the next presentation. So do we have questions for Frederick? Online or here? All right. One question for you, Frederick.
Thank you, Frederick, for a wonderful presentation. I was just wondering, you mentioned 50% reduction. Is it a question of fluidity or?
You're
I don't know the
breaking.
fluidity of your
I can't hear you clearly.
You can hear me now?
Yes, you can. Yeah, you can try.
Yeah, so I was saying 50%. So the genes that we are affected is to organize share. So my question is a question about fluidity, so maybe annotated Wait, I don't know how many copies of genome you analyzed, so let's go on that.
Okay, so I couldn't really get you, but I've heard you mention 50%, and I remember mentioning 50% at the end of the presentation. So maybe I can talk about the 50% that I spoke of. So when you look at, for example, the genome size of the camel kid, it is almost 130 MB. Then for the closest reference that I was using for the Glossina, it ranges between 300 to 366 MB for the different species that we are seeing. So this is almost less than 50%. Then we ask ourselves, what could have led to this? One of the possible features that you could look at is the number of the percentage of repeats that we are having. So for our camel kid, we are having about 15% of the repeats against what we are seeing on the closest reference that we can use, which is having about 30%, about 27 and to 30%. So that is one of the possible reasons why the genome could have been reduced to that level. And also there are other reasons. For example, you look at the percentage of completion. It could be that we haven't assembled the whole genome because it is 95% complete. We haven't assembled it, the whole of it. So that could have also led to that. But the genome for Glossina, most of the species is almost at 98, 99% complete. I hope I've answered some of your questions, though it wasn't clear from my side.
You have answered the question. Thank you.
Okay, thank you.
All right, thank you so much. So we'll go on to the next presentation, and we are going to be hearing from Dr. Francis Omonje. Dr. Francis is a senior scientist and a research leader in entomology at the NIAB in the UK. He joined in September 2021. His work bridges cutting-edge scientific research with accessible public communication. His career spans 4 countries, the UK, Canada, Kenya, and Tanzania. His focus involves in academia, national and international research. So prior to NIAB, Dr. Omonje won the prestigious Royal Society Clare Award, researching insects infecting virus genomes at Isipe in Nairobi and at the University of Cambridge. Dr. Omonje is well published in plant sciences, entomology, and genomics, and he authors 52 science stories. 52 science stories is a public space which
various research, the space of biodiversity. So maybe you'll be able to check it out later. It's a LinkedIn newsletter that simplifies complex scientific concepts. So this platform focuses on biodiversity research, especially highlighting genomics tools like high throughput sequencing and environmental DNA, that is eDNA, from the global south. So he's also an alumni of Cambridge with a PhD in plant sciences, and he's a tech founder. So today he'll be speaking to us about viromes in plants and insects and applications, hypothetical applications from his research. So welcome, Dr. Francis.
Thank you, Cecilia. Cecilia forgot to mention that I'm also an alumnus of JQuat for both my undergraduate and master's. Okay, as we wait for the presentation. Is this a plan for me to give a story? It's succeeding. Well, maybe I can just mention that as Prof. Ant said, that when you do your research overseas, you try and keep it relevant to what's happening in Africa. You'll be glad to know that I did my PhD on common bean and was working on a local variety known as Wairimo. Wairimo beans are a permanent feature in your githeri, but they're also very susceptible. It's a paradox of life that at times the tastiest products are the ones which are most susceptible. And since then, that very humble bean has produced PhD. That's myself and a colleague from Zimbabwe. And while I looked more at plant-insect virus interactions, she looked more at plant virus pollinator interactions. One of the more interesting things about that more recent work, which is to that in the present pollinators, even when the plant is infected with viruses, there is 100% recovery of the seeds, which means you don't lose any yield. Now, for the people who work on bean, they will know that bean is mostly self-pollinating, but then that shows that there's a role for pollinators in common bean in causing recovery for yields. And that is the end of my story. You have it, okay? All right, so I'll be talking about virus genomes from plants and from insects. But before I get there, I will give a brief background of the work and to contextualize how we got around to looking for these viruses and why they were important to us. So next slide, please. So we start there, and viruses have got a very bad reputation, as you may well know. And that very fitting description by Peter Medawa says that a virus is a piece of bad news that is wrapped in protein. And I'm sure with the recent pandemic, many people don't agree. But pandemics don't only occur within humans or within the health space alone. They also occur within plants and within even animals. And more recently, you remember there was a big threat to the Kenyan ugali due to maize leaf necrosis, which totally devastated the maize growing fields. And it wasn't only in Kenya, it was in Ethiopia and I think Tanzania and the neighboring countries. That happened quite a bit. But some have been around for a long time, like for the cassava viruses, which, of course, there's been concerted efforts by many research organizations to try and get for this. Next slide. So I'm going to compress, what do I press? I'm going to give you a bit of a context on what happens in the field. And there is a very interesting interaction between plants, the insects that transmit the viruses, or any pathogen, because it can also occur in bacteria, and how pathogens are able to manipulate that interaction. So in the field, and I'm going to use the example of aphid. When an aphid is flying around at a potential host plant, several cues which determine its orientation. This could be organic compounds are perceptive of these different stimuli. When they land, they do make a quick assessment. For aphids, they normally probe into the plant. They will sample the contents that are in the phloem, and if they find that to be, fitting, then they will settle in for long-term feeding, and they can feed for a very long time. And for those who know aphids, they're like Russian dogs. They are born pregnant with their grandchildren. And so very quickly, you can find that the explosion of these numbers of aphids can be very, very quick and very, very fast. But also within that, several decisions can be made. They can disperse without feeding. They can reinitiate foraging on another plant, and they can disperse feeding. And viruses are able to manipulate this host plant behavior for their own transmission. And this is really done in our lab in 2013. And most of this work was done in Arabidopsis and done in tobacco. But essentially, the crux of the matter was they discovered that there's a change in the volatile organic compounds that are emitted by plants, which are attracted to aphids, but the plant is distasteful. So in this case, there's a change in the plant stream, and there was an increase in level of glucosinolates, which are bitter in taste. And so the plants do not, the aphids then do not settle on the plant, and they opt to migrate, and then goes on. How many people know, and how many? My job was to, and in this case, it was common bean, and hence bean as a crop which we knew we can take these results and then bring them back to Kenya.
There were species-specific actions, for example, all of this for slide. But if you keep on mosaic virus, which is really understanding you will find that it has got a repulsive effect on aphids. And when you look at a principal component analysis of the protein organic compounds, this small smudge in blue is nuclear mosaic virus. It classes differently from the other two viruses.
So there are changes which do occur when plants, viruses, and insects interact. So it's impossible to conduct research whereby you disclose all these components if you really want to have a proper understanding of what's happening. But where does genomics come in? And this is a smoking gun. So most plant viruses are transmitted in a non-persistent manner, quickly acquired when the insect meet to another plant. And if you look at this micrograph from this paper by Nancy, that's a GFP-linked cord protein, and this the proboscis of an aphid. So it's really stuck right there, and they bind just loosely enough to pick it up and loosely enough to next plant. And so this is what we were interested in when we decided to use high throughput sequencing to find this virus. What else would we find of genomics? And we did. And so we changed. And I'm going to introduce you now to dicystroviruses. So this paper here is what kicked it all off. So we pivoted. So in addition to finding the normal plant infecting viruses, we also noticed a very interesting set of viruses. These were insect infecting viruses. And it was a first discovery for aphid lethal paralysis virus and big soups in bean in Kenya. And a bit of a disagreement about calling it big soup because that's an American name. And yet when we check in GenBank, ours were the first whole genomes of this virus. And this sequencing was done at ILRI, so we did not ship out any samples. So these are indigenously sequenced samples. Why they are interesting is because these viruses, they don't cause any pathology to the plants. They do not replicate within the plants. In fact, what they do is they utilize the plant vasculature to access the foraging insects. So in a way, it's as if the plant is booby-trapped. And they cause varying levels of pathology to aphids in a species-specific manner. You find that for some species of aphid, there is severe pathology. For some species of aphid, there is enough pathology to cause them to be disorientated and more susceptible to predators. And in some aphids, absolutely no effect at all. So we decided then that this deserves more investigation. First of all, to understand the diversity of these viruses within the Kenyan bean maize ecosystem. And secondly, to begin to develop the molecular tools for us to study downstream what happens when you have these viruses.
And a typical farm in Kenya. And all this is completely. And we found pretty interesting things. So we found mostly these three viruses, the digital river virus, AfricaBP virus, and a virus very similar to the 55 online virus. I don't like that name.
So while
this is very
useful for comparison, it has nothing to do with Hubei, because this particular Hubei 55 was isolated from spiders, and we were working with aphids. So when we get around to publishing this work, we will then rename it appropriately to reflect its provenance, which is Kenya. And they're different. The interesting thing with dicystoviruses is that they've got two cistrons, and the two cistrons are translated independently. So there are two internal ribosome entry sites, one at this end and one right here. And if you look at them, they are different. So for example, for BSRV is around 541 nucleotides in the IRS, and it's 197 for LPV. They seem to have different structural proteins profile. There are three right there for BSRV, there are four here. And here, I suspect is a case of ribosomal slippage. Will require a bit more work to see what's in there. But these insects are ubiquitous, and they're all over, and they cluster different phylogenetically, quite clear species demarcation in all of them. So that was step one, of the work. Step two was the creation of a full-length clone, which we did, and then all this stuff in -80 freezers somewhere. But this is the hypothesis. So this is what we're used to. Typically, you have got this plant virus insect interaction using cucumber mosaic virus as an example, where the aphid feeds on this, finds them on virus-infected plants. It picks up the viruses, but the plant is distasteful. And so you have got this cycle of transmission happening. And what we wanted to investigate using the cystoviruses is, can we break this disorientation of the aphids? And also, furthermore, the role of using POS for entomovectoring of viruses. There is some evidence to suggest that for the insect-affecting viruses that affect aphids, they are not the same which affect pollinators, but we wanted to use high-throughput sequencing to get evidence that that is the case and they don't cause any pathology, because there'll be obvious questions. If you introduce this in the environment, are you going to harm the beneficials? It's impossible to do that by any other tool apart from genomics tools. I'll then move on to virus genomes from plants, and I'm going to talk about endona viruses. So again, it started like this. The plan was to look at the diversity of known plant viruses within bean ecosystems in Kenya. Instead, in addition to finding them, we also found these viruses called alpha endona viruses. Again, these are very interesting. They don't cause any pathology within the plants. They have evolved evolutionary within the plant system such that they can only be vertically transmitted. You cannot inoculate them across or use a vector to get them across. So it's a mother, to child kind of, transmission. So this was an interesting finding, which we decided to pursue, and we got funding from Cambridge, Africa, and the Royal Society to have a look at this. And our first step, which we asked ourselves, was, what is the diversity of these viruses within the common bean varieties that are available in East Africa? So we did not look at the resistance profile of the beans because we know there's a lot of breeding efforts over the years. We just said what there. And we found that 17 varieties grown around East Africa do have endonaviruses within them. Some of them have got only one, Bacillus vulgaris endovirus 1. Most of them had got 2, and some of them had got 3 different types of endonaviruses. So again, we're able to get the whole genome information for these viruses using high throughput sequence. The first report for Africa. They had only been previously reported from bean varieties in Brazil. So questions about how we found out to investigate. What do they do in there? Now, there's 2 ways to go around this. The first one was what we did, and which I'll speak about, looking at the interaction between the different endonaviruses with each other. But the second thing, ask what if they're not there? And that will take creating fencing. Being notorious for that, it's not very hard to work with that. And so the second part has been done in pepper because pepper is much more amenable to this kind of techniques. But for the first part where we looked at the interaction between endonaviruses and each other, we noticed that they do not interact with each other. The presence of one endonavirus does not affect the accumulation of another endonavirus. There was a slight variation in the level of Bacillus vulgaris endonavirus 2 when one was present, but it was not significant. We then looked at the accumulation of pathogenic viruses in the presence of endonaviruses. So we worked with bean common mosaic virus, bean common mosaic necrosis virus, and cucumber mosaic virus. For bean common mosaic virus and bean common mosaic necrosis virus, we couldn't really make a determination. The reason is because the plant breeders have done an excellent job of breeding in resistance into most of the bean varieties that are currently grown around East Africa. And so that genetic background prevents us from really being able to measure the effect of having an endonavirus and having this pathogenic virus, because the plant background does matter a lot. But with cucumber mosaic virus, we notice there's a slight increase in the accumulation of cucumber mosaic virus. So probably they're not so good. We don't know for sure, but that's the trend, sort of look like. So that's where we reached, and we published, these 2 papers. They're available. They're open. So if you wanted, you can always download those. And the fact with viruses that we're looking at their vectors and seeing whether the presence of viruses then affects the behavior of the insect vectors. But where do we go from here? So these are just some of my propositions with this work. I find it very, very interesting and very, very fascinating in terms of the different questions we can ask with whole genome information. So for example, for all the viruses that we now have the whole genome information, we are able now to look at specific components of the genome and determine their contribution to the kind of phenotype or the kind of behavior that we observe in the lab. So that's the value of getting the whole genome. But I suggest that there may be value perhaps in having an African insect virome project, beginning to look what is within the insects that we have around, both pests and beneficials. And can this be leveraged, for example, for biocontrol or for other capabilities that possibly we can exploit? We need to create detection tools for this, and that's the only way that we can be able to do further downstream experimentation. The obvious first step is to begin developing of full-length clones. If we're able to get infectious clones, that would be much better. But then we still need to find ways of detecting this so that when we deploy them, then we are able to really experiment and find out what we can do with that. All that work is pending. We do tools for the dicystal viruses, which I developed. The rest, there's a lot of work to be done. Lastly, I think there's a lot of potential, even without having to go the really high-tech way, to begin just to look at ways in which we can begin to leverage the different viruses for the disruption of the interaction between plants and their pests. Virus research, in as far as crop protection is concerned, stalled years ago with baculoviruses. And since then, there's not been any significant progress anywhere in the world. So this is an area which I think would be keen to look at. And that's it. Those are my contacts. I'm open for collaborations around molecular entomology. As Dr. Cecilia mentioned, I'm very big now on environmental DNA, metagenomics and molecular plant pathology. Before coming here, I just come from Ghana, whereby we were looking at using eDNA to look at pollinators in cocoa. So there's a lot of work which we can do within these ecosystems. Thank you. I appreciate your time.
Thank you so much, Dr. Wamonja, for that good talk. Excellent talk. Let me elevate it further. So, and thank you for traveling to Kenya to be able to deliver that talk to us. So before we go to the next session, I'd like us to take questions for Dr. Wamonja, either online or here. Are there any questions for him? All right.
Thank you very much, Dr. Sharif, for that good presentation. You spoke about remission of seeds when you have these plant pollinators. My thoughts and what I was just thinking when you were speaking about remission of seeds is, yes, we could get the remission of seeds to increase the yield, but were you able to look at the effect of either seed transmission of the viruses? We may have the remission of seeds, but what is the possibility of the viruses being transmitted in the seeds themselves? Thank you.
Well, thank you for that one. It wasn't by direct work, but I can speak to it. There was an effect. No, for one of the viruses, there was a reduction in seed transmission. I think it was being common sick virus. I can send you the link to the paper. Then that will be more helpful. But what was extraordinary about that finding is we hardly ever look at pollinators in common bean. So we always assume that beans will self-pollinate. And so we don't even bother to conserve the environment around that in as far as ensuring that pollinators do thrive within bean ecosystems. Secondly, which pollinators are we talking about? Because honeybees will not pollinate bees. They can't pollinate, common bean. The nature of bee pollination on common bean is strip pollination. So it needs a heavy pollinator, which when it sits on the petals, then the reproductive organs do, pop out. And so we'll be talking about carpenter bees in this case. Again, carpenter bees are understudied in this country as pollinators, and I doubt even if there are any conservation efforts for these pollinators. But so the work done, it was done at Cambridge in the field. We used the bumblebee, which could be equivalent, and the common carder bee. And the results are astonishing because there was 100% recovery, irrespective of whether the plant was dying of viruses or not. That finding cannot be ignored.
Wow. All right. I do have a question. You mentioned something to do with pest management using biocontrol methods. What does the space of innovation and development around that look like? Maybe could you speak to that a little bit?
Right. So it's very interesting. One reason why I think that the use of viruses in biocontrol stalled is the nature of the viruses which are used. So baculoviruses are DNA viruses. They're more stable. You can essentially grow them in caterpillars, put the caterpillars in a blender, spin that down, and you have your, in quote unquote, natural pesticide. But for RNA viruses, it becomes much more complex. RNA viruses are more susceptible to UV light. They degrade much faster. And so then you require a multidisciplinary approach, probably with people who are working with nanoparticles. and different delivery systems for RNA viruses. So there's a lot more work that needs to be done. But
all right, we have one more question online, so we'll allow Tobias to go, and we request that you keep it brief. So Tobias, go ahead.
Okay. Thank you, and good morning, the audience. Thank you, presenter. You've done a good job. My question relates to control of diseases. Given the relationship between these plants and viruses, is it possible to develop a system where these plants will not be a conveyor of a vaccination, that is producing anti-diseases using the relationship of plant and these viruses? Have you thought in that direction? Thank you.
I think whoever is going to develop that will be very wealthy when it works. Yeah. So yeah, there's been a lot of thought, not for crop protection, but for example, there have been transgenic plants which have been developed, which release E-beta pharmacin, which is an alarm, pheromone as a way of, deterrent against, insects. So there have been attempts along that area. That wasn't very successful because after some times the insect becomes acclimated to the emissions, as it were. But yeah, there may be, room for either inducing resistance within the plant, which I think would be probably the way forward, or if the presence of the virus then changes the plant biochemistry such that the insect finds the plant to be a non-host, then there is more potential along that line.
All right, another round of applause for Dr. Monje. Thank you so much. All right. Now I'll invite our next speaker, who is Miss Riva Erasmus, who graduated from the University of Stellenbosch in 2009 with a BSc in Molecular Biology and Biotechnology. She has a diverse background in lab work and has gained extensive experience in the research field in food chemistry, stem cells, immunology, and molecular biology. In 2014, Riva transitioned to a role in sales area manager at Inkaba Biotech, who is one of our partners here. She managed sales and client relations across various areas in South Africa, covering Eastern Cape, Western Cape, Northern Cape, Free State, and Mauritius. So as of March 2024, Riva has taken a role of product specialist at Zymo Research. This is a brand that is being distributed by Inkaba Biotech in the sub-Saharan region, and she continues to apply her expertise and knowledge in the field. So
good afternoon, Sarah. Who's going to do the slides? Okay. So yeah, good afternoon, everybody. Thank you for the introduction. I am Riva Erasmus, and I am the product specialist for Zymo Research. And they are, as she mentioned, still being distributed by Inkaba Biotech. And the reason for my topic today is at the end of this presentation, I would like for you to hopefully see that there is something that we can offer to help you with your research, because I believe everybody here in this room has done a DNA or RNA extraction at some stage during their workflow. Thank you. So about Zymo Research, I'd like you to introduce you a little bit and give you a bit of background on them. So they were founded in 1994 by Dr. Larry Jar. That is the gentleman on your left in the picture. And he first isolated yeast DNA, and that is how Zymo Research started. That was in 1990. And at the top, you will see there is a picture that says room 32. And this is where all the magic started with Dr. Larry Jar and isolating yeast. And therefore, this year also marks Zymo's 30-year anniversary. And their mission is to provide innovative, high-quality products to researchers. Product lines include various tools like sample collection devices, extraction kits, cleanup kits, but we'll get to that in a bit. And the company is also known for its commitment to quality, innovation, affordable kits, and their commitment to you guys, our customers. Last year, they already had 9 subsidiaries across the world, including the United States, as well as Europe and Brazil. Next one for me, please. So they also cover 120 plus countries around the world, and they have over 80 distributors worldwide. Next one, please. And yes, today we will only be looking at the Africa region. Next one, please. So as mentioned, Incarb Biotech, just one back, please. So Incarb Biotech is a current distributor in the sub-Saharan region. They have been in partnership with Zymo since 2006, and the Incarb Biotech group of companies has close to 150 employees. They have offices in South Africa, Nigeria, Ghana, Tanzania, and right here in Kenya, in Nairobi. In 2022, Incarb opened some other branch offices in Cameroon, in Senegal, as well as Uganda. And therefore, you can see that Incarb covers the whole of sub-Saharan Africa, including East, West, Central, and Southern Africa. And in the middle, you will see there is a picture of our head office based in Makelneek in Pretoria, in Ting. Just one back, please. So that is the Zymo research workflow that we'll get into a bit. As mentioned, yeast was the first to be isolated, and plasmid isolation kits, therefore, is their bread and butter. Then there's sample collection and preservation devices, DNA RNA extraction kits, as well as cleanup kits, and then epigenetic products and services, as well as custom projects and solutions. And latest to their portfolio is the microbiome portfolio. So they are a comprehensive provider for genomics products. The first section is the sample preservation, DNA controls and reference material you will use throughout the work to assess bias in your workflow, from collection to library prep to sequencing. Then, as mentioned, the DNA and RNA extraction kits and cleanups and host depletion. This can be done if you do have a robot in your lab, like a T-can or a thermo. Such a lot. The on the right, you'll see behind there, it's also NGS library prep kits, but that's okay. We don't have to go back. I believe that all good science No problem. So as mentioned, this is a stabilization solution for all nucleic acids. It's unique in its way because some buffers on the market can only keep your DNA and or your RNA stable. So you have to choose one or the other. DNA RNA Shield is the only one that can actually keep DNA and RNA stable. And it does this at room temperature, which makes it exceptionally unique, especially for traveling in Africa. And it keeps your DNA stable up to 2 years at ambient temperatures. It keeps RNA stable up to 30 days. And if you do decide to freeze it down to -20 or -80, it's indefinitely. It's also an open system, so you're more than welcome to use any brand of DNA RNA extraction kit with it. Of course, we'd love for you to use the Zymo kits, but you are more than willing to use any other kit that you would like. It also inactivates infectious agents like your pathogens and your viruses, which makes it safe for the end user. It also makes it safe for you to travel with. So the only thing you do is if you're in the field, you're collecting maybe a stool sample or a fecal sample, you just pop it in the DNA RNA Shield tube, you close it up, you put it in your bag, you put it in your luggage, and you can ship it back to your lab. As long as you isolate your RNA within 30 days and your DNA within 2 years, the DNA and the RNA is completely stable. It is compatible with all sample types, so you can use it, again, like I mentioned, on any sample type, plants, stool, blood, any biological sample. And then this also prevents degradation from your freeze thawing cycles. And as we know, you're in Africa, also in South Africa, we do get power outages. We sometimes have a problem with electricity. We have load shedding. Your freezers break down. You lose all your precious samples. So this will not happen if your samples are in DNA and RNA Shield. So different applications, there's a bottle of the DNA RNA Shield in the middle, and that's the one that we have on the table as well. And then if you look at the bottom left, there's a buccal swab option, a blood collection vacutainer tube. Then next is the fecal collection tube. That is a little tube where if you unscrew the lid, there's a little spatula connected to the lid where you would then just scoop up your fecal sample, pop it in the tube, close it up. And then the bottom right is a saliva collection device. So the patient would spit into the tube, you disconnect the top funnel part, you put the tube back on, you invert a few times, and your sample is completely stable. Next, the toilet seat. But I think you guys know what that is for. And then at the top right corner, that is the latest innovation to the portfolio. That is to replicate a piece of toilet paper. It is a 10 by 10 centimeter square with a strip. And again, I'm sure you know what to do with that. And then once you've taken your sample, you take off your little strip and you put it in the fecal collection tube, and your sample is stable. So what is microbiomics? And the reason why I want to discuss this today is because this is exceptional for environmental samples. And then we'll also discuss some challenges in the microbiomics field. So traditionally, microbiology relies on microbial cultures for study, but 99% of microbes can't be cultured in the lab. So microbiomics or metagenomics uses NGS technology to study the microbial communities without the need for cultivation. So looking at the challenges, what are we facing here when it comes to microbiomics? Current protocols and methods have not. So the workflow is complex and it's prone to errors. And what does that lead to? Poor data reproducibility. And that could lead to a lot of bias in your entire workflow. So the complete microbiome workflow from Zymo Research. So as we
Today, the internet has decided to be tricky. Pardon us for that.
in West to Ethiopia, Burundi, but we have it in Kenya, Uganda, Tanzania, hospitals as well, within forensic labs, regulatory labs, within also yes. Then we also have another group called the CSD, or Clinical Sciences Division. That's where we have our solutions for next generation sequencing. At the same time, we also have solutions for human identification. This is where you do your forensic identification. This is where you do your paternity testing. This is where you do caseworks, and all this use the fragment analysis equipment. So in terms of sample preparation, basically at Thermo Fisher, we cover the whole workflow for molecular biology. So starting with sample preparation, we can either do automatic or you can do manual sample.
So good afternoon, everybody, and welcome back to the afternoon session. We are delighted that the conference is going on so well, and I can see many of you. At this particular juncture, we are now going to receive some briefs on the practical sessions that are going to be carried out around Africa, around East Africa. And we can start with any of the persons who are available online. Professor Antoine Mafuila, if you're able, you can tell us about the practical that you're going to do.
Yes.
Online, are you able to unmute yourself and give us a brief in about 15 minutes about the practical session that you are going to do in University of Kinshasa, and the title of the practical is NGS Data Analysis with Galaxy Tools. Professor?
Okay, I will share my my screen.
Is unmuted. Oh, yes, then.
Prof, are you able to unmute yourself, Professor Mafuila?
Yes. Okay, let's start.
So we have an echo from your side, Prof. Maybe there's another machine that's connected also in your room. You can perhaps try to use the one account, but go ahead. We can hear you loud and clearly.
Could you see my screen?
Yes, it's visible, and we can also hear you quite well.
Thank you. So in Kinshasa University, in the framework of the Africa BioGenome Project workshop, we will have a practical session on the NGS data analysis with Galaxy tools. Slide. So we will use this tool that is a web-based application that help to biologists to analyze data coming from sequencing process. So in that practice session, we will use data coming from NGS sequence tools for Illumina. So we will use this tool for analyze this data and go through the pipeline of genome assembly. So the data we will use is the sequencing data coming from the sequencing of Agaricea lamapi, that's the coral. The sequence machine is Illumina NovaSeq 6000, and this is the reference, the ID of the data we will use and we will take from the repository, and we will, with that, we will, with the practical here, we will go through different stages, going from the first queue data load into the Galaxy tools, the quality check, and after that we will do the genome assembly with this data, and after we will do the genome quality assessment, and we will finish with the genome annotation using the same tools. That will be what we will do tomorrow, to Thursday, and in Friday. Thank you. Thank you, Professor. I finished.
Roland, are you, sorry, not Roland, Prof, are you sharing your screen? We can't see the screen from our end, and we can't hear anything. Professor, University de Kinshasa, is everything okay?
Yes, I finished.
Thank you very much for that. Next online, we have the group from Cameroon. If we have Roland and Professor Liebert, you can make your presentation now. Thank you very much. We have the practical that will be carried out in Ethiopia. I see Zeudu Idea is online. And the practical that is going to be carried out in Ethiopia is the NGS data analysis from genome mapping to visualization. Thank you very much. It's visible. The slides are visible. When you're ready, we are ready. Kindly check whether you're muted. Kindly check whether you're muted and then put your slides on presentation mode.
Hi, Zedu, kindly unmute yourself. Zedu, kindly unmute yourself. We can't hear you.
Hello. Hello.
Now we can hear you. Yeah,
Yeah.
good.
Yeah, sorry.
No worries. Thank you.
Okay, that's good afternoon, everybody. I'm Zodu from Ethiopian Bio and Emerging Technology Institute. I'm one of the organizer of the practical session in Ethiopia. And this presentation, I will try to just give a brief about betting, and I will try to also highlight and really what are this the progress in NGS data analysis and its possible application, and specifically and what we are going to do really in our the coming 3 days practical section and just that analysis workflow. And I also try to highlight who are involved in this and the organizing of this training and how we selected the participant for this training. Yes, as I mentioned, just a young institute established in 2016, having 2 centers, the Bio and the Emerging Center. We do have over like 10 research directorates under the Biotechnology Research Directorate. We have the animal, plant, and the health, environmental, and also the genomics and the bioinformatics directorates, which I'm heading, and the reverse technology. We have the nanomaterial and reverse and computational science. Our major mandate is, to advance bio and emerging technology research, and we also focus on the capacity building, specifically, providing training for master, PhD, and the researcher in the areas of bioscience, data, just experimental design, and bioinformatics. And we also provide an advisory role for the government and for non-government institutions. And where I'm heading the genomics and the bioinformatics directorate, basically we are very active in conducting short-term training in bioinformatics, as you can see from these pictures, and we really collaborate nationally, regionally, and internationally, and to provide bioinformatics training for our researcher and students. And over the last 6-7 years, we have trained more than 300 individuals in the biostatistics and bioinformatics and experimental design. And we have also just some wet lab services. We know that through the advancement of NGS technologies and the cost is getting reducing, and this reducing cost has really provided opportunity really for like the continent like Africa, which is already endowed with huge biodiversity, to sequence its genetic resources. And we know that this NGS sequencing have a wider application, maybe from the genomics to transcriptomics and the proteomics, metabolomics, and epigenomics, and that this all are really a great opportunity for research in the areas of genomics. And I want to really specifically focus where really this NGS holds the potential to revolutionize African or very global agriculture. It can be used really to harness genetic diversities between and within the population, and also that it will help us also really to identify genes and novel mutation and the pathways, which are very important, maybe to improve and to accelerate genetic gain in just farm and crop planters. And as the theme of this workshop is biodiversity conservation, and that this NGS data has a huge, enormous application in the case of to assist conservation of biological genetic resources. It can really help us to estimate the inbreeding, which is very prominent in the wild population, and also it can help us to monitor the status of this biodiversity resources, and also related to EDE, thus to monitor what is really the status of especially like the fish species or the microbes. And saying this, and NGS data analysis workflow, we know that NGS data type mostly we have DNA sequence and RNA sequence, and they mostly these are sequenced by Illumina, BGI, and the PacBio and Export Nano. And mostly this data, they came in the format of FASTQ. And for this, we need to have really a quality data for downstream analysis. The downstream analysis will depend upon the quality of the data we have. And this, maybe the poor quality or low quality of any data maybe arise from the quality, the material. That means either the DNA or the RNA we use for sequencing. And also need to be very careful in the proper library preparation. For instance, like cross-contamination can lead to poor quality read sequence, and also the technical issue. And all these issues should be just carefully accounted in the downstream analysis. For this, we need to have a very key important quality control metrics. The yield, what we obtained from the sequencing and the read analysis, like the read language, the GC content, the adapter content, all these are indicators of really whether our sequence data is quality or not. And the other is the Q score. And all these have to be just critically just assessed and evaluated. And for this, we we do have a number of tools which are already developed. And among these tools, the FastIQ is one of really the well-known tool for quality control. And we have also other emerging quality control tools like the nanopod and PyCog-C. And after we observed the quality of our sequence data through just FastIQ analysis, and we need to trim some of really maybe the pairs which are low in quality, and also we need to remove the adapter. And for this, we do have different tools. That is very common and also open access. And the first thing after we obtain the quality rate, we need to map this sequence into the available genome reference. We know through sequencing, we produced a number of fragments of DNA sequence, and this DNA sequence have to be aligned or mapped into the reference sequence to to detect what is the deviation between our sequence and the reference sequence. And for this, we have well-established mapping tools from really the BW1. This is we commonly use this method for mapping, and this MWA has also different algorithm depending upon really the type of the sequence we have, like whether we have short or maybe long reads, whether there is gap in the alignment, and especially the BWME the most preferred for Illumina sequence. We have also the Minimap too for long read, like the sequence generated from PacBio and nanopod. And we need to also, after careful alignment, we need to call the variant. In the calling of the variant, we need to really assess what are the deviation from the reference genome, what are the SNPs and the indels or copy number variation. And for this, we have specialized tools like the GATK tools. These are really, in terms of like calling variants, very popular and proven. And after maybe calling, we need to visualize really this is the cause. And these are simply what we are going to implement in the coming just 3 days for our training. We need to really, for quality assessment, we need to use we're starting to use the FastIQ and for trimming and filtering, chemomatic. And after that cleaning, we need to go for mapping. And for mapping, we can employ a different tool, but the most common one is the BWA. And we need to also convert this, the sequence data into like SAM or to be BAM file. These are the most really suitable file format for downstream analysis. And we need to also mark, duplicate, and sort realignment. We can use for this the Pika tools. And for variant calling, as I said, we are going to apply the GATK and SAM tools. And for annotation, we'll use the ANOVA or Steve. And organizers, as I said, we have three individuals, including myself from Ethiopia, and Dr. Helen from Addis Ababa University, and Professor Kassan Tesfaye from Bio and Emerging Technology Institute, and myself from just Beijing and from Genomic and Bioinformatics Directorate. And this training will be a 3D training. This training will cover hands-on and practice for NGS data analysis, including from the quality control, trimming to mapping, and the quality assessment after mapping. And it also really, we try to also cover how we can really visualize some of the results. We hope that after completing this training, the participant will have the required or the basic skill, how they can handle really their own NGS data. And these are the training program for the coming 3 days. And we have around like 111 applicants for this training, and we selected 15. And this 15 just really representing 10 higher learning or academic institution and the research institution from Ethiopia. When we see the gender distribution, we have like 5, we try to include like 5 females and 10 males in this training. And mostly they are PhD students and researcher, because mostly the PhD and master student, they have been in dire need of this capacity building, because these individuals, they are going to work in the areas of data, and they have to have the basic skills to run their own data. That's why we focus mostly on PhD student, and as I said, mostly they are from from universities and followed by research institution. And the resource person, we have just trainers from Addis Ababa University, and also from my institute from Betin, from Bio and Emerging Technology Institute. And we have also a resource person from another university, from Arabamich University. These are, the brief view of our just training or workshop venue. This will be hosted by Betin at Ethiopian Bio and Emerging Technology Institute. We do have the required high-performing just computers, and we have over like 20s. We have enough space to run this training. And I would like to thank the African BioGenome Project really for working on the level base to capacitate the African really researcher and the students able to really handle their own NGS data that will literally contribute into the regional manpower. Thank you, dan asante sana. Hello.
Thank you very much, Dr. Zwedu. I mean, it's amazing that you had 111 applicants, and from that you only needed 15.
Yeah,
That should,
15.
the demand that we have for these practicals all around the region. So next, we will have Joel Ogwang, who will give us his presentation on the practical that's going to take place in Uganda. Dr. Joel, please, you have the floor. Joel Ogwang from Uganda. You are running a practical session, and the floor is yours. Okay, so I'm not sure where Joel Ogwang is. Therefore, Tresor, please mute yourself.
Hello, I'm here. Joel is
Oh,
around.
Joel.
Yes,
Please go
please.
ahead and present your practical.
Okay, thank you so much.
Thank you.
So I'm just going to share my screen.
Yes, try from your side.
Yes, yes. Yes, let me know if you can see my screen.
Yes, we can see your screen.
Okay. Yeah, so thank you very so much, everyone. Thank you, the host and everyone. My name is Joel Ogwang, and I am presenting from the National Animal Genetic Resource Center and Data Bank that is in Entebbe in Uganda. So I'm going to give a theoretical background on the practical session that will take place here starting tomorrow until Friday. And I want to talk about the following. I will introduce you to species invasion, and then I will give you a case study of invasion following the opening of the Suez Canal. And then I will discuss why COI is a good marker for studying genetic diversity and population structure. And then therein, I will discuss some concept of DNA barcoding. To begin with, invasive species are those which are non-native, therefore they are introduced through trade and travel. You find that some of these species are economically important. However, some inflict enormous economic losses. I mean, economic and actual ecological losses. As you can see from the map here, those are some of the routes through which species invasion occur. And to highlight the economic loss, economic and ecological loss due to invasive fish species, you can see that they are in, in excess of millions, And a case in point is this fish, which is called Pteroi volitans, and this one feeds on herbivorous fish species. And of course, that will undoubtedly result into algal bloom and with other consequences like diminishing coral reefs in the marine ecosystems. One characteristic of invasive species that you may need to be aware of is that they adapt very quickly. Therefore, we can trace this adaptation to positive selection of some adaptation genes, as well as to epigenetic modification of certain genes. The case in point I want to highlight is the invasion across the Suez Canal. This is in the canal connecting the Red Sea and the Mediterranean Sea is in Egypt. So you find there are species which migrate from the Red Sea, now represented well here by the Red Sea and the Gulf of Suez. You'll allow me to put my laser. Yes. And those which move from the Red Sea to the Mediterranean Sea are termed Lessepian migrants, whereas those that move in the opposite direction are called anti-Lessepian migrants, after the guy who constructed the Suez Canal. So these are some of the species that move, always suspected to be moving in those directions. So the aim here generally is to analyze the genetic diversity within and among these invasive fish species. And one thing you need to know before you can do this analysis, excuse me, you need to be aware of how to identify these species morphologically. There are very many ways to do that. You can use the fins, the body coloration, the head shape, and so forth. Those are details that we are not going into here, but you need to be aware. Then the next thing you may need to be aware of is the concept of DNA barcoding. This relies on a specific DNA sequence, normally referred to as the barcode. You find that this, in average, this barcode sequence, its divergence within the same species is really low. That's called intraspecific sequence divergence. But then this divergence increases as you go from species to genera and to families. So this is this concept alone is very important, because if within a species you find that this sequence divergence is high, then that is an indicator that probably this is a mixed species. The case in point here is COI, which is cytochrome C oxidase subunit 1, which is a preferred barcode for animals. For some reasons that, it is short, around 650 base pairs only, and therefore have primers relatively, readily available to amplify the region. And this region is also relatively conserved. Another factor is that it's a mitochondrial gene. Therefore, it is mostly maternally inherited, and they are very abundant in the cell. Of course, as you know, mitochondria are very abundant in any cell. So there is ease of getting this DNA containing this mitochondrial gene, COI. And then another factor is that I need to remove this. Animal mitochondrial DNA evolves faster than the nuclear DNA. Of course, that this makes sense because nuclear DNA, which are the classic genes that we know, which code for proteins, have to be stable. They don't. Any kind of change is quickly reversed to maintain the integrity of the genes. So this is a simple scheme to show how the pipeline, the barcoding pipeline works. We have this area where we are doing tissue sampling, DNA isolation, and PCR. And then you go into the other area whereby you have the curation of these barcodes and then eventual storage in different databases. We have BOLD for barcode of life database, or we have GenBank. And from here, many people can access, including researchers, public domain. We have the Encyclopedia of Life, and then we have industries. For example, in food industry, they use this technology, the DNA barcoding technology, to control for mislabeling. This is very, very important for sea-based foods. Just to highlight that, you may be selling a fish product and you label it as salmon. You sell it very expensively, yet it is a herring. So, DNA barcoding can help to identify that mislabeling. There are many tools that you can use to do the analysis, the molecular analysis in this pipeline. From when you have your Sanger sequences, you can process them using fragment merger and then mega, and then you can do the alignment with MAF or even mega itself. Mega, depending on the variant you are using, Right now it is 11. You can do phylogeny with different tools, including Mega itself, and then you can do population structure using a combination of DNA-SP and PopArt.
Sorry,
So
Joel,
case one.
you have Joel?
Yes,
you have
please.
about 5 minutes.
It's fine. I will
Apologies.
finish in 5 minutes.
You have 5 minutes.
It's fine. It's okay. I will finish. Thank you. So I have case one in point where we are, I'll be talking about the endemic terror species. You find that using this technology, you can analyze the sequence divergence as well as the phylogenetic relationships. And in this, when you need to be aware of the model, the best model that will give you the best tree. And normally, these have the highest log likelihood, which is a negative value. So you can see how your sequences are clustered. And then you can go ahead and look at the population structure using this network. And I want to highlight this. You can see that there are very many maternal lines here compared to this population. And when we do the practical, you will understand that this is the case of founder effect in population genetics. And then the next thing is case two, which is Serenus cabrilla. We can also see the phylogenetic relationships. We see the Mediterranean clay, which is distinctly separated from the Gulf of Suez, which is the Red Sea clay. And when you look at the structure, you actually find that there are only 4 haplotypes, that is the maternal lineages in the Red Sea, and that no lineage from the Red Sea or the Suez are shared, which means these two populations are very separate. There is no mixing between them. So inclusion, conclusion, when you analyze COI, you're able to identify genetic diversity. You can identify some cryptic species. You can also be able to clarify misidentification of species, and you can study population history events using such terminologies as the founder effect, isolation by distance, etc. I want to thank you, and if you have any questions, I'm here to answer them. Thank you.
Joel, thank you very much for your presentation. If there are any questions for any of the presentations we have received so far, you can ask them now. I can see some hands up online. Please unmute yourself and ask question. Fico, go first.
Okay, thank you, Professor, to give me this chance. Before just I'm going to raise my question regarding the offline training. I have a little gossip because I can say we are not likely due to financial shortage to be engaged for this offline practical. I think most of all those attending offline interested in this training. For some bioinformatics tools, I hope the expertise can just help us by doing so or online, because I hope they can share the training so online. Why don't you help us by doing this? I kindly request you this one. And the other one is my question for Dr. Zaudu. Did sequencing started in Ethiopia, DNA or RNA, and protein sequencing? Is it are they started in Ethiopia? I want to know this one. Thank you very much.
You want to know whether there are any sequencing activities happening in Ethiopia?
Yeah. That means is the activities started? Did they establish the sequencing institution or organization that do sequencing?
Dr. Zudu, you can answer that. Well, I have seen a lot of sequencing publication coming out of Ethiopia. I'm very, very much aware that Betty has sequencing facilities. They have some equipment that can do short read sequencing. And so I am aware that the technology is already there in Ethiopia. But Dr. Zudu or Dr. Helen can also confirm further.
Yeah.
But yeah, please go ahead. Someone was saying something. Okay, maybe ethics training happening online. It's true when you are beginning something, it's important to first do proof of concept. So the first bioinformatics training happened last year in December at the International Livestock Research Institute. Because it was on site and your facility move around, we were only able to get a few people on site. I think going forward, that is a good thing that we can begin to implement. And Joel Ogwang is here. He has heard, we have also heard, as we begin to expand our sites, we will begin to consider this. But remember that the facilitator is the one who determines the number. So you can't have everybody there, but you can have a good number. And we think for effective transfer of technology and number of maximum is a good number to start with. So, but thank you for that feedback. We have taken it into consideration. And now we can move on to the
So we invite Dr. Peter Thega to come and give us the next talk.
Okay, thank you very much. Good afternoon. Good afternoon, Gen Zs in the house. Okay, so, my name is Peter and introduced. So I'm the genomic application specialist at Eppendorf Scientific, and here I'm also representing Illumina. So for those who don't know, Eppendorf Scientific is the Illumina's channel partner in Kenya. So assist in distributing Illumina products and providing also sales support and after sales support. So my presentation today will focus on, there's a lot that has been presented on people who already have done sequencing, but I thought it's only fair for those who want to do a sequencing project, they understand what it takes. Thing that you look in your NGS workflow is the library prep. Okay, next slide. So definitely when it comes to Illumina sequencing, the sequencing process occurs on the flow cell. So in essence, we need to prepare our DNA fragment to be sequenced. And this is what we refer by library preparation. And then after that, we need to read the basis of our DNA fragment. This is what we refer by sequencing. And then at the end of the day, we need to get insightful information out of this data. We need to perform data analysis, okay? So when you talk about nucleic acid extraction, the nucleic acid of interest will affect all the subsequent steps. So in NGS, what we normally say is garbage in, garbage out. So this upstream process is usually very important because it impacts the data quality or the data that you generate at the end of the day. So what I normally advise is spend time to really think about this part of your sequencing project, okay? The other thing is, of course, you need to know or to understand what is the source that you are extracting your DNA or your RNA material. Is it genomic DNA? Is it cell-free DNA? Is it total RNA? Is it metagenomic? So these are the things that you should really think about in this step of nucleic acid extraction. Another important thing is purity of the sample. Of course, after you do your extraction, we need to remove all the contaminants that might affect our downstream processes. So it is important to know how much also the nucleic acid we have, because if it is degraded, these things can affect, or these factors can affect or influence our next steps. So when it comes now to the library preparation overview, the key things that you really look at here is what is the library preparation method, which I will highlight in the next slides. The other thing is how to choose the right library prep method for your project. What are the steps of library preparation? Which library quantification and QC you perform? And also what will be your pooling strategy? Remember, in sequencing, we don't sequence once sample. It will not be samples and sequence. So you need also to think about what is the link strategy for that. So when you talk about library preparation methods, there are so many library preparation methods out there. Performing, is it de novo or resequencing? Of course, de novo means you don't have a reference database, okay? Resequencing means you have a reference database for what you're sequencing. Are you doing RNA-seq sequencing? Are you doing shotgun metagenomics? Is your interest to discover sites of DNA methylation? In other words, are you doing bisulfate libraries? Are you doing customized libraries? Are you doing Apricon libraries, for example? This is where you do targeted library preparation for when you're interested in specific genes or variants. Or are you doing enrichment libraries? So this is very important for you to think about before you come to us and start telling us we want to sequence, because what tends to happen is the right library prep kit to use. So what Illumina has done, it has this specific tool, we refer to it as a library prep and array kit selector. So using this particular tool, you can be able to sort of determine or narrow down to the right library prep kit for your project. So what you normally do is just type online library prep, Illumina library prep and array kit selector. And then you see these different tabs, starting with the area of interest of your study. And let's move next. So you see in this area, there are different areas of study of interest. For example, is your area of interest agrigenomics, or is it cancer research, microbial genomics? Of course, you select the area that is of your particular interest. Once you highlight that, then you go to the application tab. Next, okay. So in the application tab here, there's different application methods. I created all these applications for you to be able to select the right as per your project. Okay, then we go to the method. Are you doing 16S metagenomics? Are you doing targeted DNA? So project, and then in the species tab, you also select, okay, next slide. You also select what are you working on. Are you working on maize, for example? Are you working on human species? Are you working on rights? Again, you select what is of your interest. And finally, there's a system. You select which is the system that ideally would fit your particular project. So in a nutshell, this is a very important tool to take advantage of when you're designing your sequencing project. Next to go, what are the steps of library preparation? Of course, in Illumina, we try short-range sequencing. So ideally, you'll need to fragment your DNA into specific size for sequencing. So we normally refer to that process as fragmentation, where you fragment your DNA and also you add your adapter sequences. After that, then you ligate the adapters, and I'll talk about the adapters briefly. So these adapters, you ligate them to the ends of your DNA insert or DNA fragment. And this is the process of preparing your DNA for sequencing. So you might decide to use a PCR amplification method to do your ligation, or PCR-free method. It's up to you, depending on the goals of your study. So in essence, when you talk about a DNA library, this is what we want to end up with. We want to end up with a DNA fragment containing, number one, sequences that bind to the flow cell, the P5 and P7 oligos. Of course, you want to demultiplex, or you want to be able to identify your samples at the end of the run. We refer to that as demultiplexing. So you need sequences for that. We refer to those as indexes. And then, of course, you need the sequences or regions where your sequencing primers will bind. So essentially, this is what we refer to as the adapters that you need to ligate, or you need to attach to your DNA to prepare it for sequencing. Another important method is the QC method that you use to quantify your library. So quantification, when it comes to NGS, quantification is very, very important, and accuracy cannot be understated. So because this affects the amount of data going to all samples, and it's also a very key factor in your sequencing run performance in terms of data output and data quality. So we do not recommend UV because it's not accurate, the UV spectrophotometers. Ideally, we recommend fluorometric quantification, such as qubit or qPCR, especially if you're working with single-stranded libraries. The other important thing is the quality control. So you have generated your library. So it is very important when it comes to Illumina sequencing to determine whether you have been able to nail the fragment size that you have been targeting. For example, if you've been targeting a 350 library fragment size, it is only fair for you to be able to do a QC to determine whether that has been obtained. And we recommend, in this case, using instruments such as bioanalyzers, fragment analyzers, because it allows you to visualize the size distribution. And also you can eyeball to see whether you have a product or not before you sequence. Of course, sequencing is a very expensive affair. You don't want to proceed if you're not really sure whether you have a library product or not. So that is an example of a trace that you normally get when you do your QC, your library fragment assessment. Okay, so then we come to library pooling. So, okay, this is what we refer to as multiplexing. So we typically pool multiple libraries, as I highlighted earlier, because it is cost effective to do so rather than sequencing one sample. So the number of libraries to pool will depend on the amount of data we need for each library. So, and for this, okay, next slide. So for this, we have this particular tool, we call it the sequencing coverage calculator. So it enables you to know like how many number of samples or libraries do I need to pull for this project, okay? Because that, of course, will impact the number of sequencing cartridges that you buy, and therefore it is important to have this information. So simply what you do on this sequencing coverage calculator is, next line. So it contains this particular sort of parameters where you input what you're doing. For example, if you're doing whole genome sequencing, you can say, okay, you can highlight that bit, and then you can indicate the coverage that you're targeting, the duplicates depending on what literature allows for the duplicates in your results, the genome size. For example, if you're sequencing a microbe or a species of 120 megabases, for instance, and also you can indicate the number, the total read length. Is it 300 cycles? Is it 600 cycles, etc.? And then when it comes to the instruments, then you can highlight which particular instruments you're planning to sequence your library on. So what normally happens is after you submit this, which is usually very quickly, it gives you, for example, if you want to sequence on an ExEC platform, it will tell you these are the number of samples, for example, you can be able to sequence. Okay, so library pooling. So in NGS, you hear about normalization. So what is normalization and why should we care about it? So in NGS, at the end of the day, you want to ensure there's even data for all libraries within the same sequencing run. In other words, you want to make sure everything is at the same concentration. So this is very important. So in your qubit or in your fluorometric instrument, it will normally output results for you in nanogram per microliters. But for your downstream normalization, the concentration in nanomolar concentration and also down to picomolar concentration. So there's this particular formula we normally use to convert our concentration in nanogram into nanomolar, and that is by dividing by 660 times the average library size that you obtain when you did your library fragment assessment. So after that, then you can use, depending on which platform that you can design to dilute your library using trees, resuspension buffer. So there are recommendations on what exactly you should use to dilute your samples or your pools. Okay, now let's look at the sequencing overview. So another important thing to look at when you're doing your sequencing or you're planning your sequencing, where are you planning to sequence? For example, you might be planning a project and there's no sequencing platform in the country for that. So it's important that you're mindful of that. So also how to choose the appropriate sequencer for your project and how to set up a sequencing run. So of course, if your lab has a platform, you can do it there. If it doesn't, you can always look for places where you can do the sequencing. So we have this particular tool, it's called the sequencing platform comparison tool. So you can compare all the Illumina platforms and see the different specifications that it gives you for you to be able to identify which is the best for you. And these are the different Illumina sequencing platforms that are out there for you to do your sequencing projects. Okay, as I wind up, the other thing is to know how to set up your sequencing run. You need to know how you do your plan run configuration, how you prepare sequencing consumables, how you do your denature and dilution library pool for some instruments that is not necessary, it does onboard for you, and also how to load your libraries and consumables onto the sequencer. Of course, these are the different ways you can do your plan run configuration. I won't dwell so much on that. This is just to show you the flexibility when it comes to doing your run setup. And after doing your sequencing run, it's always important to review your run performance. And we have these different ways to do it. You can do it on base application or using the sequencing analysis viewer. And in essence here, what you're looking at is what is the data quality or the data output of my run, whether does it meet the specifications. And then finally, the data analysis. So key things here that you want at the end of your run is your data to be in a format that can be analyzed by the bioinformatic tools out there, and whether the multiplexing happens successfully. And then, of course, there are those next steps you should consider in your sequencing plan, what you really need to do for your data analysis. Of course, for Illumina, we do provide various applications to enable you to do your data analysis, depending on the application that you worked on. So some of the applications are free, others need subscriptions. Thank you. And before I wind up, there's a next slide, please. So if you don't mind, you can take this QR code. There's a short quiz there. And also if you're interested with the slides of this presentation, I can send you once this QR code is filled in. Okay, thank you very much.
Applause for him while you scan the QR. Thank you very much, Dr. Peter. Do take time to visit their stand there for any further questions, clarifications. They're around, they're here with us. So thank you very much. For the next speaker, I'd like to invite Mr. Dennis Kivuva of Inkaba Biotech. Dennis Kivuva is the Sales Operational Manager at Inkaba Biotech East Africa Limited. He has a Master of Science degree in Agricultural Biotech from the Svent István University in Hungary. After finishing his master's, he worked briefly at the Kenya Medical Research Institute as a graduate assistant before joining the sales department at Inkaba Biotech. So today he will be briefly introducing his colleague who will take us through the presentation. So welcome, Dennis.
Thank you very much. Good afternoon. Greetings from Inkaba Biotech East Africa Limited. I'm Dennis, as I've been introduced. I would like to start by saying that appreciating what AfricaBP project is doing and announcing that since the last time we had the first regional workshop, at the time we were announcing that we were about to open a lab in our East Africa building, the new building. Now the lab is up and running, and we are open for trainings and practicals. So you are all welcome to visit us at any time. Without further ado, I'll introduce our genomics application specialist, Akley Moila, who will give us a brief overview of the PacBio platforms. Thank you.
Okay, good afternoon,
right.
everyone. Can
Thank
you hear me?
Yes, we can hear you.
Okay, good. Can I make just one request, please, that I share from my screen? I've made a few changes on my slides.
Yes, sure. You can
Okay.
share.
Thank you. Thank you. Thank you. I hope everyone can hear me. As Dennis has introduced me, so I'm based here in South Africa at the Illumina Biotech headquarters in Pretoria. And I'm just going to pretty much give you an update. There's been a lot of social media updates from our side, AfricaBP as well as UNISA. So I'm just going to briefly give you an update of where we are and what we are able now to do with the new platform. And I think at the end, if I have time, I just want to show you the data that we produced yesterday for one of the AfricaBP sample. Just to let you know how the matrix works and what type of data we're getting. So some of you would have seen recently, we did Africa BioGenome Challenge, Africa BioGenome Challenge, where we received a few projects and motivations, and then we had to, the AfricaBP Community Selective Committee had to sit down, kind of select which projects are more kind of would go into the voting based on criteria that we would have set, and we put that out on our social platforms, and then we finally had a winner, and this is the projects coming out of the here in South Africa from the University of the Northwest, and then they just want to sequence the horsefly. So please, whoever is speaking, can you mute yourself, please? Okay, anyway, and then also okay, most recently we had an official official media statement coming from our side between UNISA and us with the announcement of the review, and that was a couple of weeks ago. And then we did some social media outreach kind of program, and this is actually the facility where the review is housed in our lab. And then of course you would have seen some. We had also visitors from the news channel that is so that you can go and view the kind of that interview there. But that's not what I want to dwell much on. So basically, I just want to touch briefly into what the review gives us and what it allows us to do. So compared to the sequel to E, with the review now, we are able to run 4 different projects at the same time, compared to the sequel where we had to run 4 SMART cells still, but one after the other. So with the review, we are able to sequence all these 4 SMART cells all at the same time. And therefore, at the end of 24 to 30 hours, we have 4 genomes, basically, 4 animal genomes at the same time. At 30 times coverage, or you can use it for any different applications. You can do whole genome sequencing on one chip, do amplicon 16S or barcoding on another chip. You can do RNA-seq. You can do the single cell on the other chip, but all of this will run at the same time because the platform has 4 sequencing independent stages right at the back here. And then you can see here, this is a SMART cell tray, and then so this is a SMART cell tray, and then you have a sequencing plate here, and then you can run all 4 chips at the same time. And you would expect to get 90 gigabytes of Hi-Fi data, which is roughly would come to 360 gigs of Hi-Fi data. at the end of the run. So now I'll explain how we get to, the system doesn't just produce 90 gigs, but it produces the data in terabases. But I'll explain a bit later how we get it down to 90 gigs also. As I've mentioned, obviously the basic technology of PacBio Hi-Fi sequencing is that we do long-read sequencing, which is of the highest quality in the market. And then we are able to sequence across uniformly across the genomes or transcriptomes. And of course, we look at a single molecule resolution, and we can pick up methylation in real time across the genomes. And this is the data that you really don't pay for. It's part of the genome's sequence data. As I've mentioned, you have 4 chips. Each chip has about 25 million wells or ZMWs. And across 4 chips, you have 100 million sequencing wells, which produce 360 gigabytes of Hi-Fi data at the end of the run, which is runtime is 24 hours. So at the end of 24 hours, you get data across 4 chips. And ideally, we want to sequence anything between 15 and 25 KB long fragments, and with 90% of the data being in Q30 plus. And then I've just mentioning how the Hi-Fi sequencing, but what are the Hi-Fi sequencing and how do we generate Hi-Fi sequencing? So basically Hi-Fi reads are produced using the circular consensus sequencing mode in the PacBio long-read systems, and this applies across PacBio long-read platforms and provide the base level resolution with 99.9 single molecule read accuracy. So basically how do we generate the Hi-Fi insights is that we take your genomic DNA, which we would have extracted with high molecular weight kits, not the standard kit, ideally. And then this will be like 15 to 25 KB long fragments. And then you prepare your libraries by annealing the looped adapters. These looped adapters have a sequencing primer annealing site and a polymerase binding site. And then once you've completed your library, you load this library complex, polymerase complex into the chip. And then basically during sequencing, this is what happens. So you get a polymerase going around the same molecules over and over and over and over again, creating multiple copies of the same fragments. As you can see here, the polymerase just goes over that molecule over and over and over again until the runtime or whenever polymerase dies. But because this is a natural polymerase and we know the nature of polymerase is not perfect, you can see, I'm not sure if you're able to see it, you can see the red dots in each strand here. These are like your random errors that polymerase would have introduced, but like I said here, the keyword is random errors. So you see these errors, red dots distributed randomly across the molecule. And this is unlike in systematic errors where a single error can be copied into the subsequent strand over and over again. Sometimes when you do analysis, think that it's a SNP, whereas it's not. It's just an error introduced during sequencing. So because the errors are randomly distributed across the molecule, when you generate the sequences of the same molecule over and over again, you will literally flush out all those errors when you create your consensus read, which we call the Hi-Fi read. And then again, I've mentioned that we are able to pick up methylation in real time. How do we do that? So basically during sequencing, the polymerase will incorporate nucleotides at a given speed. These are a natural nucleotide, but once it encounters a methylated base, as you can see here, it takes a little bit of time to move away from that base. So the system basically looks at that the interpulse duration, the time, the lag time that polymerase take to move to the next base. So we are able to pinpoint the methylation and the type of the methylation and the location of the methylation in the genome size. So when you get your sequence data, you also get a file of methylation calls across the genomes for free. So this can be another publication on top of your whole genome sequencing. Also, what we give you, the Hi-Fi reads basically allow you to do a whole lot of applications. You can do structural variation, you can do methylation, segmental duplication, phasing of haplotypes. Because you have long reads, then you literally can see where the variants are located on which loci. And then we can do SNPs as good as any short read platform. But this is the area that a short read platform will struggle to do, the structural variation, and most of the structural variation contains most of the important SNPs that differentiate one animal to the other. And then these are pipelines that we offer for free in SmartLink PacBio. You can get this for free. You can install it on your laptop or computer cluster in the institution. You can get this for free. You can do assemblies, you can do small index calls, you can do structural variation, tandem repeat segment duplication, phasing, as I've mentioned, copy number variation, as well as methylation from your desktop. And then basically you just need a BAM file, you put it into the automation. This is a click, 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 click in SmartLink. And you can do all of this phasing of analysis for structural variation and tandem repeat, pseudo genes, copy number variation. There's a pipeline called variant calling. For phasing, we have high phase. And then methylation tools, we have that pipeline there as well. So this you do for free. And then I just want to touch a little bit on the RNA-seq. We have recently introduced a new family of products called the Kinase kits, where you are able to do full-length RNA sequencing, single-cell sequencing, as well as your normal 16S sequencing, rRNA sequencing kit. And this Kinase is based on the MAS-seq method, which concatenates small amplicons or cDNAs into one large fragments to increase throughput. The Kinase increases throughput for full-length RNA, single-cell, or 16S sequencing on PacBio platform, which enables large-scale studies at a resolution difficult to attain with short-read platform. And this basically allows us to do, usually when you do RNA sequencing for expression analysis, for instance, you want this number of millions of reads, and you're only sequencing whether the ends or the start of the molecule, and you do counting. With PacBio is that we sequence the entire transcript as it is. So number one, there's no assembly that you need. But now with this application here, we are able to generate millions of full-length transcripts. So you can literally do transcript counting from real isoforms. And this is how we do it. We take, if you're generating cDNAs, you can take cDNAs from different tissues, concatenate them, and generate one long library, and you load that into the platform. And at the end of the run, the system will do deconcatenation for you. So I've mentioned now, like this takes out the throughput of the smart cell, Revio smart cell into 100 million output, depending on the application. So you literally don't need to do short reads, RNA sequencing. If you want to have a full view of the transcript architecture, you basically can do that with the Kinase from PacBio, but also it allows you to do the counting because you have the numbers now. And what is also important, what is coming up, that we will be able now to do, and I think this is something that can be applicable to Africa BioGenome Project, where we can actually do multiple omics from a single chip. You can do genome sequence data. You can get methylome, as I've explained, but now you can also get chromatin epigenome, and then you obviously can do transcriptome with Kinase from the same chip. So you can 4 types of omics from a single chip. And if you remember, you have 4 chips per run. And then just a list of samples that we are currently running for AfricaBP. It's a whole list. We just got some few last week. And of course, we do Hi-Fi whole genome sequencing, and that is followed by Hi-C or OmniC library prep and sequencing. I quickly just want to jump out of this just to show you the parameters of the last week's run that we did for AfricaBP, if you don't mind. Actually, I'm just going to share this as well. Yeah, so basically we ran, this was a bovine genomes, one of the genomes that we got. So basically the number of bases that we produce, you can see here, it's over a terabases of data. Imagine if you had to store that from each and every run. But the nicest thing, as I've mentioned, is we sequence the same molecule over and over again, and we generate what we call the Hi-Fi reads. And how do we do that? We basically take these raw reads here, polymerase read, and then basically process them for Hi-Fi, for CCS, and basically you collapse that amount of data in terabases to merely gigabases. And this is what we expect. From a single chip, you can get 90 or at least 90 gigabytes of data. And you can see here we got almost we got over 93 gigabytes of data, and this is already more than 30 times coverage that is required from PacBio, AfricaBP genomes. And you can see here the mean insert size. So we have 18 KB long fragments that we sequenced, and the median bases were 17. And then the Hi-Fi N50, 18 to 19 KB, and then you can see the quality here, Q33. And then that means 91%. Almost 92% of the data are all in Q33 basis, which is really, really good. And at minimum, each fragment was sequenced 7 times. So that is really, really impressive from the data that we run. Of course, I can't show you all of them, but this is something that it's really, really impressive, and the review is able to do now for us at much faster speed, even much more high accurate data, because now we can stack sub-reads together and create one consensus, but we can still take that consensus and stack it with other consensus in the same molecule to generate a very, very near perfect read. And this is where you see the improved Hi-Fi accuracy. And of course, we'll be running any more samples this week as well. So I just wanted to show you the parameters and run matrix of what we are able now to do. Basically, we can sequence on average any 3 gigabase genome. We just require a single SMRT cell. And as I've mentioned, we can produce the true data that we produce is in terabases. But imagine if you had to store that from every single run, you need really, really great storage infrastructure. But no, we don't store that. We take that and the instrument on board, it process that data to compress the files, generate Hi-Fi data, and compress that files to really in gigabases range from terabases. So in a nutshell, that's what I wanted to share with you. I'll be happy to take any question from here.
All right. Thank you very much, Acclaim, for being patient with us and waiting online, knowing that we're running a bit late on our schedule. So we also want to thank INCABA so much for partnering with the AfricaBP project on the agenda to sequence 100,000 genomes in 10 years. And we have a lot of other partners here that we also appreciate. So thank you so much, Acclaim, for that good presentation. And because of time, I would like us to move to the next presenter. I'm sure Dennis here will be able to take on any questions that come from that presentation. So thank you very much. So our next presentation is from Mr. Yoichi Shimada, who is the CEO and founder of CA Medilinks Kenya Limited. Yoichi was engaged in developing sales and marketing strategies for pharmaceutical companies and medical device manufacturers at ZS Associates, a US-based strategy consulting firm. He also participated in the launch of Afri Inc., now Senri, a logistics IT startup in Uganda and Kenya. While studying abroad in France, he saw the medical situation in East Africa, which was far different from Japan. And he realized that there's a business opportunity in the healthcare and life science field. So he holds a BA in law from University of Tokyo, an MA in public policy, and he's a graduate of School of University of Tokyo. And he will give us a talk today on CA Medilinks and the opportunities that they present for partnering with Africa BioGenome Project and all of us here today. So, Mr. Yoichi, welcome.
Hi, good afternoon.
So Mr. Yoichi will be connecting his presentation shortly. We'll be able to begin his talk in a few minutes.
Good afternoon again. Yep. Right. I'm very happy because I can see your faces who would be motivating the research field in the future for the future of Africa. And then I'm also happy that I can be a part of that. All right. So today, I want to talk about our companies and our services. And finally, I want to introduce the concept of innovation hub. Two things which I want you to remember throughout my presentation. Only 2 items. Number 1, you don't have to ship your sequencing samples all the way to the other countries. Why? Because we have locally available solutions for sequencing. Number 2, when you want to start off your new research activities, CA Medlinks, based in Nairobi, will support you, regardless of the field, such as human research, animal research, or even plant researches. We are here to help you. Only 2 items. So let me get into some more concrete discussions. So let me introduce our corporate visions. Perhaps you might wonder why, as the only Asian, I am here, right? And then you may feel that why Chinese guy is here. Actually, I am Japanese. I may look like Chinese, but I'm Japanese. All right. So I have been in Kenya since 2018. I wonder why. I want to bridge the gap for healthcare and also life science innovations. I'm coming from Japan, where people are enjoying the longer lifespans. Perhaps you may be hearing about our countries. For example, like people here are having the average lifespan of 84. A good news is for women, it's much longer. Now 88. Unfortunately for men, only 81. But how did we achieve this? This is through access to healthcare. And then this is also through the access to life science innovations. A lot of Japanese researchers from public to the private sector have been striving to find new solutions for disease areas such as cancers or any other non-communicable disease or even infectious disease. That's sustained by the investments, the research field. Here in Kenya and the other African countries, from my experiences, from my exposure to the market, that system is not necessarily there. So that's something which I want to contribute throughout my company services. When I was talking about this company, CA Medlinks, CA. CA stands for Connect Afia. So what is Afia? Afia is from Swahili word health. So what we are seeing is 2 aspects. We want to connect you are charged more. The turnaround time is longer. You are not sure about the quality of the samples. These three major challenges, we want to resolve it by giving a local solutions. So this is the range of services which we are providing. If you are interested, our team, our technical team is here at the booth. You can talk to us. Okay. Yeah. We also we want to work researchers as a prime topics, and even collaborative projects. We are open for that. Okay. Now, I want to talk about this innovation hub. So this is what we want to show you. We want to introduce you as main agenda of today. This is our commitment. However, they repeatedly complain that they saw the equipment at the public institution level. They saw Illumina sequencers, they saw MGI sequencers, but equipment and also have chance to promote their ideas to the other parts of the world.
En fonction de l'usage,
This is what we want to achieve as
l'usage
innovation
du
hub.
document.
Why we call it hub? Because we want to coordinate between these local researchers and the global stakeholders. So we have 4 pillars. We have working space in our lab, which researchers can visit and then stay, and then do their own work. We also provide some of the networking events. Last year, we had 3 events.
Même 15 et demi. Si c'est pour le concours. Comment travailler le process? Comment? C'est il va le délai. C'est au cas de 2 semaines. 1 à 2 semaines. Le concours, c'est quand? Non, quand je mets une à deux, ça veut dire que le temps que l'hume, ça veut dire cette semaine, quand vous m'avez saisi, donc que je passe par les informations à faire de sa disponibilité, il faut lui donner au moins une semaine. Si lui aussi fait faire, ça c'est pas le même qui fait. C'est fabriqué.
The research process. For example, if you want to screen the people, you might expect that to screen people may have access to the written archives. We also want to help such researchers to publish their research results. And I really want to push the publishing potentially and we also want to get the sponsor to our activity so that we can sustain these activities. So this is what we want to achieve. So going back to the introduction, two items. I hope you still remember. I have forgotten. Just one. So we are, you don't necessarily have to zip up your sequence samples when these are samples all the way to the other one. Number two, even enhancing your research result. Throughout this innovation house.
Ok, bon. Merci beaucoup.
Thank you.
Ok. D'accord.
Thank you very much for that presentation. I hope you're getting a picture of the opportunities that exist within reach. Yeah. So CMA Links is here with us today as well. They have a booth, but I'll allow one burning question. Just one? I think they'll come to the booth. All right. Thank you very much. All right. So our next presenter is Miss Florence Parsimei. She's a distinguished professional in the biosciences industry with expertise in molecular biology, bioinformatics, cell biology, protein cell analysis, and microscopy. So she's currently the regional manager, Africa Biosystems Limited, overseeing the biosciences division in 9 African countries. Her work has significantly advanced knowledge and public health in the scientific field, leading to initiatives in COVID testing, schistosomiasis control, STI testing, and wastewater surveillance. As a life sciences consultant, she provides strategic insights to enhance health programs and research projects, ensuring scientific advancements that translate to real-world benefits. Florence excels in leadership, communication, training, strategic planning, and business development, driving profitable growth and maintaining strong stakeholder relationships. Her dedication in advancing biosciences and improving public health establishes her as a leading figure in her field. So as Florence connects to the projector. Mr. Paul, maybe you'll assist her so that she can show her slides. So Ms. Florence Parsimei works with the Africa Biosystems Limited. We've already had Caroline Barire. So Florence is here to tell us about, just to give us a brief taste of what some of us will experience in the next 3 days, in the hands-on practical. So I'm sure there's some of us here who will attend that. And next year, hopefully they'll be able to create a bigger space for more of us.
Good afternoon, everyone. Good afternoon, everyone. I hope the lunch has sat in. I hope you're not sleeping because I have a lot of interesting things for you today. So as you heard, thank you, Dr. Cecilia, for that introduction. My name is Florence Parsimei. I'm a product specialist and technical specialist at Africa Biosystems Limited. And we are very happy to partner with the Africa BioGenome in this particular workshop and also conference. So as you well know, most of you have applied for the workshop in different areas. And Africa Biosystem, together with Thermo Fisher Scientific, will be hosting the practical on unveiling the cutting-edge technologies in nucleic acid extraction and detection. So tomorrow, for all those people who are registered for this workshop, kindly let's meet them as early as possible. We are really trying to start by 9. So if you can be there by 8:30, we'll be really, really happy to host you. So without wasting any more time, I'll just go right into the presentation. Yeah, so workshop is focusing on unveiling the cutting edge technologies in nucleic acid extraction and detection. And here, the objectives of our training mostly is to, the people who are going to attend this training are going to efficiently perform nucleic acid extraction and gain a comprehensive understanding of our Kingfisher automation platforms for extraction. Also, they're going to be able to grasp and articulate the principles of real-time PCR, including detailed discussions of its various chemistries. I will not go into detail about the chemistries, but these are the things that we're just going to cover during understand the mechanisms to which the instruments transform signals, and also conduct precise maintenance and calibration of the instrument, as well as diagnose and resolve fundamental troubleshooting scenarios. So you'll be able, by the end of this training, you'll be able to just cover most of the things or all of them in this particular workshop. So our main agenda for this particular workshop is to just make sure that you are familiar with the nucleic acid extractions methods, whether the manual or automated with our Kingfisher platform. So tomorrow we are going to get to see our automated Kingfisher platform for extraction. And we are also going to be able to do hands-on with our silica-based kits as well. Also, we are also going to learn about the real-time PCR principle, the different chemistries of the real-time, whether it's cybergreen or Tachman assays or probes. Also, we are going to also set up a real-time PCR experiments and also do analysis on the software. So it's going to be very intense, but it's going to be good at the end of the day. We are also going to analyze absolute quantitative experiments, things like gene expression, things like SNPs, and such things tomorrow. So also now this is just to give you an overview of what's going to happen in the lab. As you can see on my screen, we have automation platforms on my right. I have to do that. On my right side, so we have the Kingfisher platforms. Tomorrow we are going to interact with the Thermo Fisher Kingfisher Flex, the second automation platform. We're just going to check the features and probably just know how it works. And then, but tomorrow we are also going to also do the manual extractions with the GeneJet kits, the spin columns, as you can see on the very end. Someone would ask me whether we have extraction kit for people are working on bacteria, there's people are working on plants, and so many other things. So one thing I also want to mention, we have 3 ways of extraction that we support. We have the organic extraction, whereby you use we call it CTAB method. So for here, we have the trizol, we have the dinazole, which can help you to do organic extraction. Okay, sorry. The other thing, I hope the ones online can see. All right, so the other method we have is the silica-based methods, which we are used to, the kits that we are used to in the lab. We have the GeneJet kits and the PureLink kits. That's what we offer from Thermo Fisher Scientific and Invitrogen, respectively. And also another thing that we also offer is the magnetic bead method of extraction, whereby I'm talking about the Kingfisher platforms. They use the magnetic bead-based methods of extraction. All right, so as you can see my screen, click quickly, we can be able to extract from a wide range of sample types. We have viruses, tissue, cells, blood, plants, FFPE, buccal, and all that. So regardless of your sample type, we have, our kits have been specified for those applications. All right, so after you do extraction, I like going with the workflow. After coming out from extraction, the next thing you want to do is to quantify your DNA, just to find out whether you did something or, before you proceed on to the rest. So we have our qubit fluorometric quantification instruments. And here, these ones are, I know some of you have interacted with them in the lab, but for those who have not seen, these are quantification solutions that use fluorescence, and the dye binds or intercalates into the DNA, and that's how you're able to know how much DNA you have in your sample. We also have the nanodrop as well. I know for people who don't want to use fluorescence, we can use spectroscopy, which we also have the nanodrop as well. All right. I don't know whether my slides are moving. All right, so we also, after doing your quantification and you've confirmed you have something, the next thing is wanting to amplify your target to be able to know what you have in your sample. We have endpoint PCR systems, right from the smallest minimum to the Proflex. I know you have interacted with thermocyclers in your lab. For people who are doing molecular biology, we got you covered. All right, then we have the convectional PCR master mix. These are just the basic PCR master mix that you use in the lab. But one thing I can tell you about our master mixes, we are very big on master mixes. And I think for people who have interacted with our master mixes, they have never been disappointed. And if you have, kindly come and see us in the tent. Don't say it right now. All right, so we have hot start, high fidelity, long range, multiplex, GC region, direct, and broad range types of PCR, and also standard. So we have master mixes across every type of PCR that you would want to do, because this is something that has been done over many years by Thermo Fisher Scientific, and we always improve the technology as we go. As you can see, there are some 3 main master mixes here. We have the Super5 for high fidelity master mixes. We have the hot start for people who don't want to work with ice. And we also have the direct PCR for people who are avoiding extraction. So we got you covered in every step of the way. All right. Then, of course, after doing your amplification with your master mix and on the convectional PCR, we have the ones that you call it cleanup. Isolation of DNA fragments. When you want to clean up, you probably want to do downstream like sequencing. We got you covered in terms of the kits for cleanup. We have the bead-based cleanup kits, and we also have the spin column-based kits if you want to cut your gels. And you can see the type of bands that are produced by our reagents. All right. So right into the heart of it, we are going to also discuss the real-time PCR, which is our main focus for our practical. People are like, okay, yeah, we've done real-time PCR and all that, but do you know the chemistry? Do you know why you should use CyberGreen and not Tachman probes? Do you know all those things? So this training is going to answer all those questions, and it's going to also show you, based on your budget, what you can work with. As you can see, we have the other instruments that we have over here. But for our practical, you're going to use the Quant Studio 5, right? I will not go over this slide. I know it's so crowded, but it has the real-time master mixes. So all the master mix you require for your real-time PCR, we got you covered. Then the other thing is the PCR plastic seals and accessories. Of course, we have to use plastics. For high quality plastics, kindly reach out to Applied Biosystems. They do not melt in your thermocycler. You spent millions on your thermocycler and then you just find a plastic that are stuck there. So we don't do that. We give you high quality plastics based on your throughput. All right, so last but not least, I will just show you a small tour of the lab of where we are going to do our presentation tomorrow. Our workshop tomorrow. We're going to do it at the National Public Health Reference Labs in Kenyatta. They have been our partners for a very long time, and they have supported us in so many validations that we do. So I'll just play this video. Okay, it doesn't have a sound, but you can just see. Oh, sorry, you cannot see. My God. Let me see. Okay. Let me just try again. Right. Okay, I don't know why it has refused to play. Oh, I just opened YouTube. Sorry, give me a second. Okay, yeah. So this is just a tour of the lab. I was there. So I'm inviting you, all of you, to come and join us at the National Public Health Labs tomorrow, starting 8:30. I know, okay, it's not all of you. We had shortlisted but next time we are going to plan something big that is going to accommodate all of us. So as you can see, this is a tour into the lab. They were very busy. As you can see, they are always busy. So this is the sample processing room where you'll see the Kingfisher Flex that I was talking about, the automated platform that I was taking with my phone, so don't worry. As you can see in the hood, they are preparing the samples to load in the Kingfisher automated extract, which is serviced and supported by Africa BioSystem, as you can see. So this is our extraction platform that we are going to go over. Again, you can see me in the lab over here. So these are the Proflex instruments. These are our endpoint PCR systems that, of course, Africa BioSystems has been supporting and also servicing. Again, you remember in our first presentation, my colleague mentioned that we offer technical support. We have application specialists for that. And also we offer service in terms of, our engineers. So as you can see, I will not go over all this video because of time, but I just want to say thank you for Africa BioGenome to give us this opportunity to be able to talk about what we do and what we are going to do tomorrow. So thank you so much, and karibuni sana.
Great, thank you so much. You've seen what we'll be missing tomorrow. So next time, we'll rally them to have more spaces for us to be able to participate in more practicals. And also, as it was mentioned here, you all work in labs, you all work in institutions. Partner with the Africa BioGenome Project. You have something to offer. You know stuff. You can teach us that stuff. Yeah? All right, so we're gonna get another taste of, so one more presentation for the practical. So the next speaker is Dr. Anubab Khan, who works with Professor Hela. Professor Hela was here in the morning and told us about the work they're doing around wildlife genomics. And he mentioned that they'll be having a training, hands-on training in Kilifi at Pwani University. So we're going to get a bit of a taste of what will be happening there. And I don't know if you found him already online.
Hi, I don't know if you all can see my screen. I don't know.
So Dr. Anubab, if you can hear us, you can go ahead with the presentation.
Yeah, can you hear me? Sorry, can you hear me? Can you see my screen?
Yes, we can hear you and we can see the presentation.
Thank you. Hi, everybody. I'm sorry I'm missing all of this. I am in Kilifi where we are doing a workshop. We are actually doing 2 workshops, and the workshops are all happening in Pwani University in Kenya. And you can see it's on the coast. It's very close to the beaches, which is very nice. The workshops are being offered in collaboration between Copenhagen and Pwani University, and the objectives are very simple. So far you already know how important genomics is for a lot of wildlife studies. So we want to demonstrate in this workshop how specifically how genomics has been helping wildlife and conservation at ground level and at theoretical level. And we want to build a network of researchers who can stay in touch and who can support each others. Generally, what happens in a field like this is if you start doing the research and you might just be one of the very few people in your own university or sometimes even in the country who is starting to do this. And if you run into trouble, there is nobody to help you to troubleshoot the issues. So building a group of researchers who know what to do and a group of researchers who can help each other would be very good. And we're trying to build this. I've been doing this for some time in Pwani University. I've been doing it for 2 years now. And hopefully we can build capacity in the tropics. So I do workshops in several countries along tropics, mostly in Asia. And now we have started doing it in few places in Africa as well. And hopefully all of these efforts will help build a push within the country for developing infrastructure. So right now what we are finding is that the basic necessities for teaching genomics is also poor in a lot of tropical countries. I'm not talking about research. Research needs a different kind of beast altogether at times. I'm talking about basic shared facilities. So facilities like high throughput computational infrastructure, sample preservation kits, museum facilities. All of these are majorly lacking in a lot of tropical countries. But if a lot of people come together and if there is a push within the country, then maybe a lot of consortiums or governments can be moved to build these facilities. And that's one of the objectives. And hopefully all of this will stop parachute science, which has been happening rampantly across the tropics. So by parachute science, what I mean is generally it's someone from the West coming in, taking samples from your country to the West, doing the research and presenting the research as their own, while the species and the people for whom the research really matters are based in the tropics and they're not acknowledged very much so. So that's the objective. There are going to be 2 workshops. First workshop is a 3-day workshop from 7th to 9th of August. That QR code is a link to that website that I've mentioned. There are 2 partners. So I and Sammy Wambua are from Pony Universities. I'm an adjunct faculty there. And Rasmus, Anus, and Thomas are from Copenhagen University. And I am also partially affiliated to Copenhagen University there. It's a 3-day workshop, and it is majorly to give a taste of how genomics can be used for wildlife studies. And it will do some very basic training on what to do when one gets the data from a sequencing facility and estimate things like population structure or inbreeding, basic assignment tests, things like that. The next workshop is a 2-week course. It's quite intensive. It's about 80 hours course overall. It's offered again in collaboration between Copenhagen and Pony University. So Rasmus is from Copenhagen University, Sami is from Pony University, and I affiliate with both of them. Here, the goal is, of course, to teach everything starting from basic population genetics to how these things can be used for curbing trafficking of wildlife, for example, or if you have assignments to do, for example, a new lion ventured into your park, how would you figure out which area the lion came from? Or even at a policy level, if you have to do translocations, for example, from zoo to wild or between 2 national parks, what would be the best way to introduce animals or translocate for genetic rescues? We will try to assess how to measure threats to wildlife. It's going to be quite a fundamental course, and it'll start from the very basic level and move on to some very advanced levels. Yeah, that's the basic overview of the 2 courses that are happening. The QR code should give you access to the websites for these courses. Hopefully, we'll see a lot of you here soon. Thank you.
All right, thank you so much for that introduction. I hope you guys got the QR codes and look out for more opportunities. This team conducts trainings on bioinformatics from time to time, affiliated to the AfricaBP and also to other programs. So you may be able to access trainings from them. So yes, thank you very much. And those who are going to Kilifi, I hope they'll get a chance to enjoy the beach as well. So I'll invite, yes. All right. I'll invite Professor Ann to invite the next speaker.
Let's just confirm whether Professor Julian Usuji is online. So I think as we're waiting for me to look for Professor Julian to log on, this is a great opportunity for us to take a 15 minutes tea break. Just serve tea, join us in the room, and we will listen to the presentation by Professor Julian and Dr. Thangod. So those of you who are online, join us in 15 minutes. That is at 30 minutes past the hour. Okay, so tea is served outside. Maybe we can all move that way. Let's also take the time to have tea with the exhibitors so that we can visit them. And then I will call you back when we are ready for the panel.
Okay. Brother, but we wanted to like we already have removed the around. Okay. Was wanted.
So before we have the next presentation, which is the last presentation of the proposed African Open Institute Fellowship in Biodiversity, we're going to have a short panel session with young early career scientists from Kenya. So I will call up Dr. Teresia Mongai, Ms. Howie Adede, and Mr. Mike, and I'll ask you to introduce yourself and say what you do and maybe why you applied to come to this workshop.
And now we have who are supposed to check the language so that then what's up? Our approach, first of all, is what? Because first of all, what we need to work with. These people are ready. So we don't need to get.
Then good evening. My name is Teresia Mongai, and I have a PhD in ecology, but I'm currently I have a PhD in ecology where I majored in soils, contamination, and all that. Currently, I work with the Center for Ecosystem Restoration Kenya. We saw Andrew presenting here, so we work together in the same organization. I'm based in Kilifi, where I work in the coastal ecosystems, restoring mangroves, cultural forests, and all that. And yeah, that's a bit.
Yes, so my name is Michael Landy. Currently, I'm doing my PhD in cassava epigenetics. And the goal that I'm doing in epigenetics is trying to identify epigenetic variations that affect cassava yield and also towards infection to cassava mosaic disease. Yeah.
Adede Howie Nyodero, Genomics Center, next in molecular lab, and I'm going into my master's program at the University of Oxford.
So she's very shy about it, but it's Ivy League university, and we all aspire to go to such university. So when you have an opportunity, it's good to say. So the question is the journey, how easy it is, because in Europe, student having started schooling at maybe 6 years old in grade 1 attains his PhD, or her PhD is around 24, 25. You know, about my journey, I got my PhD 30s, yeah, and I think the age is coming down now. But the journey, tell us about the journey and maybe some of the challenges and not only the challenges, but even the solutions you have been able to put forward to address them.
When you sit and probably listen, I read here, or you look at stories of success scientists, you might see it's just part of
so many nice achievements that look nice on the
and it pains to be at the point where, okay, why just stop? Because funding can be a challenge at times, and because it's a challenge doesn't mean that you just abandon and move away. There are many ways to manage. So it's been quite a tiring process, but the thing that I look back every time I think about it, I never stopped and gave up in the middle. Because at the end of it, when you think about your story, then it does make sense because many people who have gone all the way to their PhDs and are even maybe in the innovation sector, like Dr. Cecilia or are still in academia, do have quite do have the challenges that this, but giving up point where people leak out of the journey. So yeah, do that.
Yeah, good story. So yeah, I think what I agree with also you is that also for me, I guess with my journey, is it hasn't been straightforward. But my bioinformatics journey started way in my undergraduate, in my third year, because I was always excited about computers, and when I could do, because my background is biochemistry. And when I could figure out central dogma in terms of codes, then I immediately say that this is what I wanna do. All through my undergraduate, the most important thing I would say, even for the undergraduates here, is to do a project at least, so that it can help you with the next step. Because at that time I did a bioinformatics project, and then left undergraduate. And then obviously, like any other African student, once you're done, you'd want to do
Thank you for that. That's quite interesting journey, and it's kind of a bit different from any other field. And that's why I think we comparison is a bit of joy. So I'll start with, I did my BSc here in Jomo Kenyatta, and I graduated in ecology. After that, I didn't stay for long. I applied for the University of China
scholarship, the one that was here. We were one of the pioneers. I went to China to do my master's. I did that for 3 years. After 3 years, I applied for the TWAS fellowship, again with the University of Chinese Academy. And I got to do my PhD as one of the first people to do the CAS-TWAS President Fellowship. And then that was 6 years I spent in China studying. And then now I came back to Kenya. I did graduate under 30, yeah, Yeah, I graduated when I was 29 with a PhD. And yeah, so from there, then we can say I started to notice the pitfalls when it comes to studying, and then now joining everything together without taking the breaks in between. And I think that's where I found my challenge, because now the challenge comes where I've taken 6 years studying. Kind of workshops where you can meet people with different experiences and coming from different backgrounds and even work and school, and you get to meet people because your network is your network. So now you know people who can link you up, who can give you information. We've had a lot of talks about grants. I think this was something that was not very accessible to us when we were studying, but here they're playing the grants are available, you just need to go for them. So when I came back, it's a skill that we had to learn, actually, how to actually write those grants. Because nobody is going to write them for you. You write the grants and you look for the funding. So that's exactly how I started my journey when I came back. I got into the online coding. I found grants for young careers, people who are young career scientists. I think we have one of them. It's called the report that supports young students, young career scientists who come into the market directly. I gave it a try first time and I got it. So that's how I found myself in conservation. Thanks.
So some great ideas from you guys to yourselves, you know. What I've heard from listening is that you have to be a person of integrity so that, you know, whoever you're interacting with can actually trust you. You have to put in the work, you know, to apply for grants. You have to put in the work to, when you get a project, don't sit around, try and work very hard so that you can be, you know, among the first people to finish, to present some results, because then it's when somebody is asked, give me one student to do this project, they will remember you, the hardworking students. But I've also heard that networking is very, very important. And this is one of the things that we're doing here in Africa BioGenome Project. We have brought in people from not only the different countries around Eastern Africa, but we have also brought you who are here from different parts of the country. We have people from Alupe, we have people from Mombasa, we have people from Ethiopia. We are all here so that you can be able to interact. So I hope you are able to walk away with one contact or two contacts, not only from the friends you have been sitting with, but also with the presentations that you to career paths, because, you know, in the career paths, you know, maybe for me, I'll push you into academia, but I don't know whether everyone wants to be a lecturer, you know, or wants to stick in the university and teach. So what are the career paths that are available for early career scientists? Or maybe I ask that question in another way. What interests you? Starting with Adi.
The world of science is very broad. Many scientists that I have met in my journey, especially in the first 2-3 years, were entirely in academia. But when I finished my BSc, I got into the field of innovation and entrepreneurship in science. It was very different, and it really took I had a very steep learning curve because yes, I'm doing bench work, but the bench work needs to translate to something tangible, something like get a product out, and this product into the market, can it benefit people? And then there needs to be like a clear communication, because you're taking a product to people who are not scientists. For them to take it up, they need to understand the practicality. So there's a lot in science, not only in academia, and what will keep you in is that interests you. Because for me, my research are in medicine, translational sciences, because I want to take my work from bench to bedside, like from the lab work into the people out there who have the needs that we are trying to sit down and research and, you know, make presentations. All the presentations that we're making here, there are some people in the audience who were making a few comments like, okay, the moment I hear all these complicated science words, I don't understand. And this is the person who you want to take the research to. So identifying what your interest is and knowing where the gaps are and what your end goal is the main thing that can help you diversify outside the world of academia. Academia is important. There's a lot of interesting and important research that's being done, but the thing that pains me the most in the process is most of the times after a publication is made, you get your credentials, celebrate, and then move on and just leave it at recommendations and walk away. But you've invested so many years looking into so many minute and important details to bring out something that you and a few other people understand really well, and then just leave it in that small niche. So there's a lot of opportunities. You just need to tap into that thing that is your interest, because working on something you're not interested in will bore you to death, and you will not move from that point of bench work out into the world. So I think there's a lot out there, and people, we who are in the field, need to understand it even outside the circles of science. Yeah. Multidisciplinary research is also an avenue for you to expand your mind and get to know that besides work agents working on the bench, what can I do? How can I tap into the bigger world that's out there? Because we're not just doing papers or just that level. We are trying to solve real life problems, real life challenges that are actually affecting real people in the world.
Nice. So I'll like to echo what Dr. Cecilia mentioned yesterday, that whatever disturbs you is probably that's what your passion is. Yeah. So I'll respond to that in two ways. So short term and also long term, ultimately what I desire. So obviously after my PhD, I would like to further do a postdoc still in plant research, basically bioinformatics, and also work in the field of research. But my long-term goal is capacity development. So I'm really passionate, especially for the undergraduate level. And why I'm saying this is in line with bioinformatics, while I was doing my master's, we came up with a group called Bioinformatics Hub of Kenya. I don't know if there's any members of that in the room. I see a few. And what we were trying to do at the undergraduate level is to try and sensitize, or sort of having an outreach events where we sensitize about bioinformatics, and also I'm a fanatic of open science. And what we realize is at the undergraduate level, students are not even aware of this kind of science that we are actually having an advantage of listening to various talks. So we sensitize it and also pick up a few students to try and mentor them. All right. So end goal is capacity development in terms of mentoring these students to also realize there's potential in this science. All right. And so that's basically my end goal to venture in capacity development. And I'm glad that I now starting to involve myself with African BioGenome. Also, I have to mention we be part of the project that is going to be in IITA because they've already started the collaboration, and also doing the workshops with African BioGenome because I mean that's where I mean impacting the knowledge, and then also for the participants who are impacted with the knowledge, I challenge you also to go out there and take the initiative to also train the people who are all either your juniors or even your seniors, and then be open in terms of sharing your experiences so that we can always have this peer learning within our spaces. Yeah, so.
Okay, so for me, I'll start with a story. So when I wrote my publication, and there's always, when you're writing a paper, there's the recommendations part. So I've done a lot of science, the soil science in the laboratory. Then at the recommendations, I wrote this is like addressed to the policymakers and the people on the ground, because my research that particular research was about a dump site that we have in Ngong. So at the recommendation was like, I recommend this and this and this. Then all my papers go with recommendations to someone else. Then later I asked myself, do these people actually read these papers and read the recommendations that I've put here? Or it's just for me and everybody in science who gets to see this? So it dawned on me one day that I might actually have to go back to my country and do exactly what I recommended. I have to be the person that goes and does and makes a difference, or teaches and disseminates this kind of information, because it might not have gotten to the people that it needed to be. I have to be that person. So of course, the dump site, I saw the paper was featured in a newspaper, I remember, and the dump site was moved. It was made into a landfill, which was also very motivating. I was like, oh, so they actually here and something can actually happen. So when I came back, even the first project I did for conservation and restoration was back where I come from. I come from Kajiado, from Ngong. So the conservation project I wrote was to empower people in my community to make a difference where I come from, because I understand the challenges that are there and what was going on. So with that project, what we did is we did capacity building as well, taught the people around how to plant trees effectively, how we put up the first nursery there in the Ngong Hills. So that was capacity and empowerment and mentorship as well, because we had to work with schools and disseminate this kind of knowledge. So I would say for me, my passion is to change, to make a passion and skill. I link that to service. So with the passion and the skill, it's for me, but the service, it's to the people, it's the community, it's to the world, it's to our country.
you can make a difference wherever you are, just by the knowledge that you have learned. It's so profound that asking yourself challenging questions. So it's not about doing a paper for the sake of doing a paper, but actually follow through because this is your passion. And then Adi said you have to be passionate. If you're not passionate about science, then you will never be able to get that scholarship. So remember that we are in Africa. It's not a straight path, but if you stay in science, you'll be able to get there, and you'll be science doing. So science is fun. Science has kept me going for the last, I've been working in this area, actually in Jomo Kenyatta for 33 years. I'm still here, earning a living, still enjoying the science. So much more for you because you have more opportunities than the rest of us who are slowly making their way out. And then capacity building. As we build your capacity in whichever area that we are building, look at those who are behind you and see what skills you can present. So I ask you to give a round to this magnificent panel. And I thank you so much for the knowledge that you have shared. Indeed, that has also resonated with us and myself, and there are many things that we can pick and implement from what you have told us. Thank you very much. We can take our seats now. Thank you. I think now it's time to introduce our online panel. We have Professor Julian Usuji.
Hello, can you hear me? Hello, can you hear me?
Yes, I can hear you. So let me introduce you as we, will you have the slides from your side or maybe you share from your side?
Okay.
So Professor Julian Usuji is a very, very active member of the African BioGenome Project. He's the coordinator of the West African node based at University of Port Harcourt. He's a molecular geneticist and he's a pure hardcore graduate. He's now a professor at University of Port Harcourt and chairman of the university-wide accreditations committee, which he served from 2016 to 2017. He was the pioneer dean in the School of Science and Computing at Clareton University in Nigeria. Professor Julian is a professor of molecular cytogenetics currently. He's a principal investigator in the interdisciplinary biological network. And he's a very, very active member of our West African node. And I hand over to you now, Professor Julian, to speak about the AfricaBP fellowships that the African Open Institute is running. Welcome, and the floor is yours.
Thank you, and greetings everyone who's listening. This presentation actually is supposed to throw more light on the effort of AfricaBP to drive knowledge exchange, which is part of our grand challenges. We all know that worldwide or globally, we have a pressure of extinction of species. And this pressure of extinction has been so troublesome. A lot of species across various environmental limits are being affected. And for this reason, a group known as Alliance for Zero Extinction has been very busy trying to locate organisms threatened with extinction and then plan strategies that could help to conserve them. And this Global Alliance for Zero Extinction is made up of 93 people from 93 institutions from 37 countries. Can you see my slide? It's not moving. Okay, thank you. The effort towards the conservation of species has been on the increase. And the latest one is the one by the AfricaBP BioGenome Project, which tries to use genome sequencing, to facilitate conservation of species. And can you hear me?
Yes,
can you hear
we
me?
can hear you.
Okay, thank you very much. Yeah. So the AfricaBP BioGenome Project has affiliate groups like Africa BioGenome Project, the BioGenome Project Canada, European Reference Genome Atlas, etc. And these different groups are looking inwards, trying to look at the organisms existing in their terrains, and then to see how to apply genome sequencing to facilitate conservation of the species there. Conservation and the restoration of biodiversity through genomics is of paramount importance to importance in genetic, conservation of stopping genetic erosion and safeguard the genetic diversity of Africa's plants and animals. AfricaBP that was established in 2021 with a goal of sequencing 100,000 genomes in 10 years, and thereby through that generated 203,000 gigabases of DNA sequence data. AfricaBP considers that the best approach is to support the sequencing and analysis efforts on the continent. Hence, there is a need to develop and empower African scientists to do this. Much of what has been sequenced as African materials, organisms, have been sequenced in Europe and America, and actually outside Africa. So part of the interest of AfricaBP is to develop capacity that can help us do these things in Africa. And then that will help us also to cover more organisms and then also have our sequence data domiciled in Africa. Well, different countries have been adopting different approaches, both in Africa and elsewhere, to get this done. And what I'm just presenting here is like the Nigeria case, where there is a governmental public effort to conserve biodiversity of object concern. And here in the map up, you will see that there is a blend of organisms. I mean, a blend of environment in Nigeria, the landscape, showing various blends of forest and vegetation. And what Nigerian government did in the past was mark out areas with concentration of species of conservation concern, which you can find as patches, dark patches in the lower map here. All these are locations of these 20 forest and game reserves you see here. Each one has its own particular blend of organisms from, in fact, all of conservation concern. Now, Africa BioGenome Project, as a way of trying to help this whole process, knowing also that Africa doesn't have the capacity, decided to implement one of these grand challenges. That's the technology development, knowledge exchange, and industrial grand challenge. Through this grand challenge, the Open Institute for Genomics and Bioinformatics was established to facilitate capacity building. And the AfricaBP Open Institute programs to achieve the capacity building include graduate fellowships and trainings. We also have fellowships for young leaders. We also have fellowships and programs for people who are actually in the service. The idea is to have the capacity polished and sharpened for greater achievement. And the fellowship package has an international exchange outlook. The fellowship AfricaBP is having has a tripartite structure with Africa BioGenome Project there. Then we have African institutions there, and then possible partners. It could be corporate partners. It could be institutions from elsewhere. So that's the kind of structure we have in the partnership. And the partnership has rules for different members of the group. For instance, AfricaBP serves as a coordinated Pan-African effort to build capacity, generate, analyze, and deploy genomics data for the improvement and sustainable use of biodiversity in agriculture across Africa. And then the partners are expected to promote homegrown and regional networks for human capital development to achieve this genomics and bioinformatics fit. The partners are expected to provide funds, and then also they expected to recognize the capacity of AfricaBP for assessing regional, manpower for the program. This is an outlook of what we have achieved so far. This first one here is with the African Genome Center, based in Mohammed VI Polytechnic University, Morocco. Here we had a program that involved fellowship for 10 Africans. These 10 Africans you see here, each person is from a different country. And then they went to Morocco. They had to learn and practice, put into use bioinformatics, and then use it to sequence an organism there. The second one under it is the one, another fellowship for young leaders that was between AfricaBP, University of Illinois, and the University of Port Harcourt. Here we have 4 USA around September. And then these ones are going to spend 4 months in all, 1 month in African lab and 3 months in the US. And at the end of the day, they're expected to go through the whole process of sequencing and bioinformatics to analyze and then get a paper out of it. Then this one at the middle is a poster for a new fellowship that is being driven by anchored by IITA. Hello, can you hear me?
Yes, we can hear you.
All right. Okay, thanks. Okay, thanks. I was getting an echo here. So each of these fellowships and each other program that we have in Africa BioGenome Project is encouraged to work towards having a publication to anchor that. Each one will prepare a paper from it that will be published in a very strong journal to serve as an evidence of our progress. And there are actually criteria for these fellowships. This particular one set of criteria is for the Emerging Genomics Leaders 2024, which is the one that will go to Illinois. First, the person, the affiliation is that the person must come from an African organization or institution. Another criterion is the relevant experience. The person's experience must be relevant. The person who's been to molecular biology, molecular genetics, genome sequencing, assembly, annotation, etc. And then the person can be at any stage in the career, depending the requirements of the fellowship. The person can be a student, the person can be a lecturer, and different levels of the academic order. And then the person must be an African national, which is very important. All right, here we have justification. Every fellowship anchored by Africa BioGenome Project must be justified. The justification, could be taken to several forms, like contribute to knowledge and technology transfer. That is one. And then the fellowship must provide support for bioinformatics and genomics capabilities, leverage on already established partnerships, and generate sequence data. Each fellowship also has a set of objectives, which means the objectives for one particular fellowship might slightly differ from the objectives of the other. For the young leaders, we targeted 20. Well, at that point, we targeted 20, but we're looking at 200 now, especially because we are already in at this level where we are now trying to massively sequence these organisms in Africa. And the beneficiaries should be in biodiversity, should focus on biodiversity genomics and bioinformatics. Will be in agricultural, medicinal, industrial raw materials genomics. Will be in the area of ethics, policy, and social issues, and then sampling and background and biobanking. Each in this whole fellowship thing, there are responsibilities for home institution of the prospective fellow, for the prospective fellow, for the selected fellow, and for AfricaBP. For instance, the home institution of the prospective fellow must have organized any of the AfricaBP Open Institute regional workshops, hosted any of the practical sessions. The home institution should also be able to nominate a candidate for the fellowship, sponsor part of it, develop a curriculum and research programs in line, as aware of bringing in or properly utilizing the fruits of the fellowship, and then cover visa processing fees for the fellows. Then the prospective fellow also has his or her own responsibilities. For instance, should be able to apply for the fellowship on the AfricaBP website, arrange with the home institutions for nomination and provide letter of support, identify the African organization where the person can do an internship, etc. And then those of the prospective fellows that are actually selected as beneficiaries of the fellowship also have their responsibilities, which are stated there. And then Africa BioGenome Project also has our own responsibility, like provide scientific directions for the fellowship, call for applications and advertise the fellowship program, shortlist and select fellows, and seek additional funding where necessary. And the funding also pattern has a structure. Here, the African institution or Center of Excellence that is involved in the fellowship should be able to take care of the home tuition fees, accommodation costs, research, and living expenses within the country when the person is still there. And then the partner outside the fellow's country should be able to bear the cost of research, living costs, accommodation, and all that research costs for the fellowship, while the fellow is there with them. Now we also have expected outcomes from the fellows. One, at the end of each fellowship, we expect that the fellows should be able to develop curriculum for genomics and bioinformatics programs in their various institutions across Africa. Develop research programs in genomics and bioinformatics in their various institutions, and also help to develop, have leadership skill that can help in further capacity building by training others. we have expected outcomes of the fellowship programs. Now, this is like a summary of the outcomes. The first fellowship, actually, we had at least 10 beneficiaries. The second one, we had more, but when we checked I showed there. And then for each fellowship, we expected to have at least one journal article, very good, strong, high quality journal article. And then the fellowship should be able to support capacity building and academic mentorship. And then more biodiversity data, yield more biodiversity data for agri conservation and biodiversity genomics. I think at this point, thank God we'll need to throw in some light. Thank God, are you there?
I think you can go on. Thank God had said he might not be able to join us.
All right, thank you. All right, thank you very much for listening. Yeah, thanks for listening.
Okay, so thank you. We had a few challenges on our end, but Kagame can be able to project some of the slides that we may not have seen. But we've been able to showcase.
Okay, I meant to project some of the slides again.
But no, we're doing it from our end.
Okay.
Good. Yes.
Okay, beautiful.
Are there any questions, Vivian? Chat?
Okay, maybe let me stop sharing so you beam it from your end, and then I go through as you project.
It's okay. I think you can read for us some of the questions that must have come up. I think for us is to be able to showcase to Africa that we are also doing our bit in terms of sponsoring fellowships. The fellowships are competitive, and your home institution has to be a partner with Africa BioGenome Project for either the lecturer, the student to be eligible. Prof, are there any questions that have been posed online?
Okay. Let me check. Okay.
I can see one. Sequencing cannot be effective in Africa unless we start developing our own sequencers, our own chemicals, and our own reagents.
Yeah. Okay. Yes. Okay. Yes. Well, the issue is, well, as a matter of fact, we don't need to wait till we're able to develop our own equipment, our own reagents, and all of that. It simply means that we may not be able to. It simply means we may not be able to sequence at all, because if you look at the sequencing machines, the simplest ones are, in fact, the very simple one, simplest one is about $1,000 US. You know, and how many people can actually get that? And it also has a limit. You can use it maybe for some microorganisms, but when you look at plants and animals with the genome sizes, you will see that it cannot be helpful. The one that can maybe do like an organism that has a bit like 1 gigabase size of genome, like the Illumina ISEC 100, that one can be used. But when you look at the general economics, hello, can you hear me? Can I go on? Can I go on?
You're clear. Let's continue.
Okay, can I continue? Can I continue?
Yes, please do.
Okay, all right, thank you very much. Yeah, so when you look at this equipment, they're actually very expensive. ranging from the smallest one of about $1,000 to the ones that are up close to about $1 million. And that amount of money is not easy to come by. And that also makes the sequencing very expensive. It is very good that Africa thinks of having their own models or brands of this equipment. But it will also require people to know what is required, know about sequencing, know what it takes to sequence, know what the data is analyzed, so that we will not be able to waste the effort. For now, what AfricaBP believes is that it is good through partnership to be able to start sequencing organisms in Africa. And it means we will use equipment built elsewhere, manufactured elsewhere. We will use consumables that can work with those equipment. And then once we have African organizations or institutions that have some of those facilities, we, through partnership, we work with them and start sequencing our species while we wait for a time when Africa will be able to produce such equipment and the consumables. I don't know if I answered the question well.
The question has been answered. Is there a question on the floor in Kenya? I wonder, from my end, I can just
yes. Somebody asked, is there somebody asked, is there
explain to our audience how it come about?
Yeah.
Is there a lab space, and how do you choose the institution where the fellowships are held?
Yeah, those first and foremost, we have lab spaces for various forms of genomics and bioinformatics works in Africa. The only thing is first to know where we have facilities and know where the capacity, human capacity is to put those facilities into use, and then be able to start using them. There are so many locations in Africa that have sequencing machines. So many have not even been used before. So, and we need to have, which is our primary target of AfricaBP, to have people trained so that wherever such facilities are existing, people trained that have been trained can help put them into use. Whether for epigenetics, genome sequencing, or whatever. Once people are trained and their equipment, then they can be really put into use. Then the fellowship question, the question of fellowship says, in addition to giving fellowships, sequencing of the graduating student samples may also be effective for sequencing African biological diversity. Well, that is true, but provided the person, the graduating students are associated with AfricaBP based on the funds that come to us. The people providing the funds have their own target specimens, species, organisms. If graduating students are working with such organisms, then those organisms can be, the sequencing project or program can feed into the AfricaBP fellowship thing. But if the person is not part of AfricaBP, the person is not working with AfricaBP, AfricaBP doesn't have the fund yet to start sequencing stuff for individuals. So that's the way we are going for now. We can do it in the nearest future, but presently, based on the financial limitations, we are not working outside our defined portfolio at the moment. Thank you.
I think it's important for us to re-emphasize that we have received requests from individuals. I'm working on this species. I am doing my postdoc in it. I want it to be sequenced. because of the goals and the objectives of the African BioGenome Project, we want to make sure that what is sequenced is a reference genome that will add value to an existing body of research. So it is difficult for us to adopt species without going in a systematic manner. So we already have plants, animals that have been nominated by nations and countries, and we are systematically sequencing those. But if we come to your institution is a partner of Africa BioGenome Project, is where the fellowship is going to be held, and there's a project that's ongoing, say, like the project that we had this morning on sequencing of giraffes. It would make sense to sequence that giraffe because it's already directly contributing to the knowledge within that institution. So we are going systematically. We're not saying no, but it has to be within the agenda of Africa BioGenome Project. The human resource available, maybe we will bring in just one person from the next village, yeah, to support, but the human capacity is there. And that is how we are going to build these hubs. So we're very, very keen to have hubs because we don't expect there to be a sequencing machine in every institution, in every city. But we want to ensure that the sequencers that are placed in, say, in JKUAT or in University of Nairobi are fully utilized, fully utilized, using our collective samples and our collective work. It is the only way we will be able to drive the economy of scale. Reagents coming into the country will be cheaper because they are bulk. The instrument running time will be cheaper because you're running it at high throughput, full. All the lanes and all the cells are being utilized rather than running a one cell on a, instead of 4 cells, you're running 1 cell, or instead of 16 wells, you're running only 1. Yeah, that way we are going to leverage on the economies of scale and we'll be able to do things in a much cheaper and in a much faster way. Professor Julian, anything else to add? So I don't know whether Prof is still online.
Yes, I am.
Yes.
Somebody also had a chat asking which effort that is being made to get more institutions to participate in this program. Yeah, what we do is we have been sending out messages. We send out these posters, for instance, and encourage people to register and be part of it. So many people have been seeing the posters and some procrastinate, they will do it tomorrow. And by tomorrow, some other things distract them, they don't remember to register. As who are in this present workshop at JKUAT, will know what is happening. And when they go home to their institutions, they take the message back. JKUAT needs institutions in Kenya, and not just Kenya, East and Central Africa, to indicate interest in joining in sponsoring this workshop. It is not easy, but if they are able to raise the fund and have workshops, the people around those countries and those institutions will benefit. And we do these workshops once each year. And it means that the people, after being there this year, will not want to miss the one of next year. And they'll tell their friends and colleagues. We post them in national, like Genetics Conference, Society of Nigeria, those national bodies, professional bodies. We post these things there so that more people will know. But we cannot force people to join or join our train. We encourage people to be with us because people have a lot to gain. Those who have been on board have gained enormously, and those who are joining will continue to gain. So, but the most important thing is we are open Our website is open. People should go and register and be part of us. People should join the different working groups, pilot committees or committees. Once people participate and they belong to national bodies, academic or professional bodies, they will post these flyers and posters there, announce these workshops there. More people will know about it and register and participate. If maybe 10 organizations or institutions participated in this present East Central African workshop this year, we are expecting that 10 times 10 should be there next year, because the message is going out, the gospel is going out. We want Africa to stand on her feet. That is what we are doing. And we want everybody to come on board. Thank you. Somebody also asked, do we have such facilities here in Ethiopia? How can we apply to the facilities of the Africa BioGenome Project? I'm currently working on resistance genes against the coffee. Okay, fine. If you look inward in Ethiopia, you'll be surprised that you have sophisticated genome sequencing facilities. Even if you don't have the most current ones, let there be old ones that can be managed. By the time you start, the result will not be top-notch. It may not be, it won't have the level of accuracy that the current present ones could offer. But at least we start from somewhere. If you check in Ethiopia, you must find a place where you have people who are in genomics or bioinformatics. By the time you ask them, they will likely have an idea where you can have such facilities. That's what I have. Thank you.
Indeed, we do have people who are interested in genomics and bioinformatics around Africa. There are numerous universities that I can name off. Like, for instance, in Ethiopia, you're asking Addis Ababa University. We have the Bio and Imagine Institute also that have this sequencing facility. So, and over and across the region. So let us support them. I think for us, I also hope that you realize that we have revolutionized the way conferences are held. Before, conferences used to be for very small number of people, but by leveraging on technology, we have been able to increase the number of people who can be available in person and also virtually. And if we have 10 institutions that are hosting, each of them are hosting 100 people, we'll be able to have 10,000 people in person just by leveraging on technology. And that is when the money will follow these opportunities, because we will start a movement, and we already have, where people will say, hey, there's something that is happening here, and we should be able to invest in it, and it's a good thing, because we are not asking the participants to pay. We are actually asking for support so people can be able to have access. But I want to thank you all very much, our online audience, our in-person audience, our conference exhibitors. We have now come to the end of the first part of this conference. It's structured in a 2-day conference-style workshop. And from now on, we will break out into groups. You will go to the institution where you have been called to go to do the practical. Now, I have received very, very many requests for opportunities to practicals. So we are working. If your institution joins us, next year, maybe we will have 100 sites where we can have hands-on training, so that the 100 of you, each of you will go somewhere and receive hands-on training. But this year we had 7 sites. The 7 sites were shortlisting maybe 10 people. So you see the numbers were limited. So, but we have your contacts. You are now considered to be part of the African BioGenome Project. I hope we shall see you in person next year, and virtually also for the next year workshop. It is scheduled to be run during the first week of August of 2025. So be around. Make sure you have added to your qualification. If you're an undergrad, make sure you've passed and proceeded to the next year of study. If you're a graduate student, make sure you have passed and now you're in the field. If you're a postgraduate student, maybe you can be like those new doctors that we had presenting their papers. So when you see the call next year, excuse me, please apply to present your poster. Also apply to present your work. And we're giving opportunities to early career scientists. So try and you will be called. We will be able to put you on our program. I want to close by saying we would not have run this program without our technology companies. All the 5 companies that partner with us right now in AGBL. We are very grateful for your support. Recognize that flew from Pwani at the coast of Nairobi, coast of Kenya. So the investment was huge.
We want to also appreciate the Director of Surgery, the Vice Chancellor of Jomo Kenyatta, for hosting this very, very important conference. We really are grateful for the partnership, to a long-term partnership with Jomo Kenyatta. Jomo Kenyatta is the host institute for the African BioGenome Project because the project needed a home, and Jomo Kenyatta is the current host. So we are grateful for that support of the Vice Chancellor. The Vice Chancellor sent the grateful for the workers of surgery who have provided time, and we are entirely grateful for even the transport department who have provided logistic support. Finally, but never the least, you would not have been able to run this project
and say we have participants on ground without yourself. So give yourselves a round of applause for coming. And the local organizing committee, okay, I'm the chair, but I will thank myself. I would not have been able to do this without Dr. Kuja Josiah, Mr. Paul Kagame. Can you stand and we give you a round of applause? Paul Kagame, Dr. Cecilia, Yves Hernandez, who we co-opted at the very last minute. It calls for lots of meetings, but with a team of 4, we were able to have this workshop. So thank you very much. I am aware that Dennis from Inkaba has a little speech to make. As one of our technology providers, we are very, very grateful for the support that we've received from Inkaba Biotech right from the beginning. The first organism that, the first set of organisms, I should say, that you heard from Yves' presentation was sequenced by Inkaba Biotech in their labs in South Africa. We're looking for you to invest in state-of-the-art Revio and one of those high-end equipment in Nairobi, Kenya. And these people here have promised that they will utilize it to the full. Dennis, is yours.
So thank you very much, Prof, for the introduction. So yeah, I was here before, so without the organization, Dr. Cecilia. So yeah, a small thing.
Dr. Josiah.
in that section.
We keep it for you.
Yeah.
Prof. Thank you very much, and we look forward to you organizing the next year's event. Thank you.
Thank you very much, and we must appreciate the team that you have come with for their support. And
Olivia in South Africa will need a picture, so I ask you that we just come and you take a picture so you can prove to him that you you actually planted the basket, but you not only more important. So we've come to the end, and I'm so happy to see that you're not running out like somebody has said, fire. And that way we can socialize, we can mingle, and we can create more networks. Asanteni sana, and we'll see you all tomorrow or the first week of August 2025. Thank you. Just a small announcement before you see the recycling bin. And if you have empty bottles behind you, you can just go and in there.
Capricorn. Yeah, they can degree. Nah, that's my thing.
Well,